And Dr. Adamala, whenever you see you have to share your screen, you can override whatever it's in there, so you don't have to wait for us to remove the slides.
Thank you.
You can kick me out.
With pleasure.
All right, Dr. Spatz, you can get going.
Okay, thank you, Temo. So my name is Joachim Spatz. I am giving the introduction of today's session. So we are approaching day 3 of SynCell, and I want to use this opportunity now. Well, I'm one of the co-chairs, but I really want to acknowledge the amazing work what Gabriel, Telmo, Jacqueline, Delora actually did, and I probably forgot now someone, but they really pulled this whole conference. And as Gabriel just mentioned, he did it completely without any stuff. So I think we really have to appreciate his power and his energy, which he put into this conference. And I think it became a great success. We have more than 600 participants being registered, and that's pretty cool, I would say. Yeah. But so before we start, I was asked to give a little bit of a perspective in the field, what's happening here. And what I want to share with you today is actually a more educational combined with research approach, which the Max Planck Society in Germany is following since a couple of years. And let me share my slides with you. And what I want to bring up to you is to talk a little bit about the idea of the Max Planck School, Matter to Life. And the Max Planck School is an idea which originally started from the current president, Martin Strattmann of the Max Planck Society, who wanted to develop a graduate school system which makes use of the scientific excellence which is all over Germany. And Germany has a very dislocated structure in terms of universities and institutes. So there are many hotspots over a rather small country, if you compare it with the US, for instance. And the idea of the Max Planck School is to combine the expertises which are dislocated, but to bring them together and use the scientific expertise eventually to train the very best international students, which we can recruit. And one of these, and there are currently 3 schools running. So there's the Max Planck School for Photonics, the other one is Cognition, and the third one, which I'm the chair of, is Matter to Life. And let me give you a few ideas about what Matter to Life, the Max Planck School, means. So first of all, the challenges, and that's how it actually started, is that we were applying for the school having a scientific and the educational topic, which is novel and not covered by any of the sites in Germany. So none of the sites which we have and is able actually to cover such the story of Matter to Life completely. So it requires the collaboration of different sites, different cities, and different universities. And so we were looking for the best scientists in Germany, research and educational wise. And that eventually then, I think, allowed us now in the third year, we are in the third year now, to recruit the best international and competitive students. So it started 2017 with an application which I wrote. You see at this time it was still called Construction Life. It changed now the title to Matter to Life, I think, which is a much better one. And we are now in this, we have just recruited the third generation of students. So what is Matter to Life? And I think it's a very research-driven educational program. So the questions, education, and the courses we are giving to very talented students coming from all over the world are very much based on challenging scientific questions from the best scientists in Germany in this field. And you can, if you ask me what is Matter to Life, what I usually say, well, in the past, there's a lot is known from living cellular systems. So it's a kind of the biophysical analytical approach, which started to really conquer the world, which became very successful maybe 20, 30 years ago. And these days, I think little is known, but there is progress, obviously, in understanding how to build a living cellular systems. And that's what mainly the topic of this conference is also all about. So that's an engineering approach. So we are moving from a more physical approach to an engineering approach. But what I think is not known and understood is how assemblies of molecules into systems make up a living cellular system. So you can ask actually, having lipids, bringing them together, when do these assemblies actually make up life? And there's really a fundamental knowledge gap. So there are fundamental questions you need to ask, but which have also very strong consequences for applications. So Matter to Life is all from molecules to cellular systems, at the end to decision-making systems. And I think this decision-making is something which makes up a living system. So it somehow autonomously can make a decision to go this or the other fate or pathway. And here we have to look into engineering molecular complexity. It's about selectivity and it's about non-equilibrium structures, which are obviously very important. And the consequences is here that we have actually to develop a new integrated scientific engineering and also ethical approach. And especially the last one, is the ethical program, is something which is really important here to have, because for a non-expert, it's obviously a question, what is actually matter to life besides scientists? And coming back to the idea of having really a dislocated structure, and you see here Germany, and with a couple of hotspots, and now I marked only the ones, universities and institutions which are part of the program. And we are connecting all these places by a very professional digital, so video-based interface. So the lecturers and the students do not need really to travel to hear the lectures, and that works pretty well. And that was developed actually before the Corona pandemic. But of course, we benefit a lot by having this in place when Corona eventually started. And I think we have also the very best scientists recruited from Germany in this program. And one mark is, of course, the Nobel Prize, Stefan Hell. He's very well known. He's very engaged in this program. But then in Germany, there's the Leibniz Prize, which is one of the most important prizes, or is maybe the most important prize in Germany for science in all different fields of science. And this is a snapshot of the fellows. We are a faculty of 50 professors all over Germany, as I said, and they are all engaged in different activities in the school. And more importantly, I think, is that we have only a few numbers of students. So we have, let's say, 3 hotspots of the school, which is one in Heidelberg, where the focus of the lectures is in chemistry and engineering. In Göttingen, the second one is in complex systems and biological physics. And bioengineering is located in Munich, the Technical University of Munich. And each site only accepts up to 10 students per year. So it's a very competitive program. And it's not so simple to enter, but I can say that the quality of the students is really excellent. It is a great pleasure to work with them. So we have 50 faculties, and each year only up to 30 students, which makes a very nice ratio between faculty and students. And that allows a very close interactions between students and professors. The timeline is so that we are accepting students after the 3rd year in the European program or the 4th year program in North American, so with a bachelor from all different fields. That can be material science, biology, mathematics, computer sciences. And we have an excellent preparation course, which allows us to somehow bring all students up to a rather similar knowledge level. And then these students, within 2 years, they get their master in matter to life from these universities. And then if they qualify, they enter into a 3-year PhD program. So after 5 years, the students will have a master degree and a PhD degree within the Max Planck School, matter to life. The students you see here, again, a snapshot of, I think left is the 1st generation, right is the 2nd generation. That's when Corona started. Obviously, we are then coupled by Zoom, not anymore gathering as on the left side, but we hope to come back to this. And at this, I'm also at the end of my introduction about the Max Planck School Matter to Life, I think, which is a very important contribution to develop the science, actually, which lies behind it, because it's based on the education, and it relies on the education of the students, which are the future researchers, scientists in this field. And with this, I thank you very much. I'm not aware if we should ask question now, but I guess not. And with this, I hand over to Ilya Platzman, who is now the chair. Ilya is a very well-known scientist. He's part of our department, and he developed very nice concepts in synthetic cells the last 5 years, I would say. And so Ilya will take over and will introduce the speakers. Thank you very much.
So thank you very much, Joachim, for your kind introduction of me. Then I would like to welcome everyone for the 3rd day of SynCell conference. And let me immediately start with introducing the first featured speaker for today. It's Kate Adamala. She is a synthetic biologist and a professor of genetics at the University of Minnesota. Professor Adamala received her master's degree in chemistry from the University of Warsaw, Poland, studying synthetic organic chemistry. In grad school, she worked with Professor Pierluigi Luisi from University of Roma and Jack Zotzak from Harvard University. She studied RNA, biophysics, small peptide catalysis, and liposome dynamics in an effort to build a chemical system capable of Darwinian evolution. Kate's postdoctoral work in Ed Boyden Synthetic Neurobiology Group at MIT focused on developing novel methods for multiplex control and readout of mammalian cells. It is important also to mention that Professor Adamala is a founder and steering group member of the BuildACell, which is an initiative and international collaboration for creation of synthetic cells. Today, Professor Adamala will speak about lineage agnostic biology. I'm looking forward for your talk.
Thank you. Thank you so much for the introduction. And thank you to all of the organizers for keeping this conference going. I'm really glad that we're able to meet. Too bad it's not in person, but I think it's been really fascinating seminar series and a really interesting last couple of days. So thank you very much for the opportunity to talk today. And what I want to talk about is an overview of 2 example projects that we're doing in our lab in an effort to reconstitute as many natural biological systems in a synthetic cell as possible. And then at the end, I will take a few minutes to introduce the BuildACell initiative, which I hope everyone who is here, who is interested in synthetic cells, would also be interested in joining, in working with us, in collaborating with us. So just to get started, all of us have different motivations for getting into this field. For me personally, as a chemist, it really appeals to me that we're able to build biological systems from scratch, that we're able to control where all the molecules go. But kind of at the big picture rationale for why I think this is the absolute best field to be in is the evolutionary perspective. If you look at the life on Earth, all of the known terrestrial life comes from this single last universal common ancestor. So essentially all of the biology experiments that's been done up to date were done on a sample size that equals 1, because we only have one modern terrestrial life, and that's all biology has to work with. So to me, the biggest rationale of our field is to expand that tree of life, to be able to build new lineages, to be able to experiment with what can happen. How can life be done in different ways than the way life is done right now? So basically build lifelike systems that expand on a current architecture, and then eventually, we're not there yet, but eventually one day build a completely separate tree of life, build a living systems that don't come from this standard last universal common ancestor lineage, but instead be based on different kind of biochemistry, different kind of principles, find a way to do Darwinian evolution in a different way. So this is why I find this the most fascinating field, and I hope I share that with at least some of you. And I think this is why this is the field with so much promise and so much, we're seeing so much interest and development right now. So just to kind of clear up some definitions out of the way, when I talk about a synthetic cell, we don't kind of have an agreement in a field what exactly is a synthetic cell, but to me, a synthetic cell is a biochemical system that has some emergent properties. So it's something that is more than just the sum of its parts. So it's not a simple biochemical experiment, but it's still fully understandable and most importantly, fully controllable. So we know exactly where every gene goes. We know what every gene does. We know what every molecule does, and it's not a black box like a regular living system. And to be more precise on a molecular level, to me, a synthetic cell, and that's how we define it in my lab for practical purposes, it's a liposomal bioreactor that expresses proteins from DNA genome. So we have a liposome membrane that encapsulates cell-free protein expression system. And that's the basic architecture of a synthetic cell. When me or someone from my lab says we're working on synthetic cells, this is exactly what we're working on. And the membrane is not that interesting. In our case, membrane is most of the time a lipid bilayer membrane that is made of phospholipids and cholesterol, just encapsulating that cell-free protein expression system that's inside. And so this general architecture, as shown here, is what the synthetic cells that I'm going to describe in both of the projects that I'm going to focus on today are based on. It's a bacterial cell-free protein expression system. All of the projects that I'm going to show today are based on E. coli cell-free expression system. And we're using DNA genomes to express all of the genes. And then before I get started into specific projects, I want to show this one of my favorite slides that's hopefully proving why this field is the next best thing after sliced bread. There is this incredible variety of applications and feedback between studying natural biology and building synthetic cells. If we build tools for making a cell from scratch, those same tools can be applied to understanding complex natural biology, including healthy and disease states of our own cells. So there are immediate, direct biomedical applications. There are practical metabolic engineering applications in biocomputing, in creating new tools for studying natural cells, but also applications in kind of a fun areas like astrobiology, like studying the origin of life, the earliest evolution of life. So it's a very varied field, and that's one of the reasons I'm so attracted to it, and I hope all of you are so attracted to it, because we're biochemists, we're working on very specific trackable problems, but the applications are nearly endless. Everything you can do with biology, hopefully one day you'll be able to do with synthetic cells. So now let me introduce two projects in my lab that both kind of the unifying thought behind most of the projects in my lab, most of the synthetic cell projects, is we want to reconstitute natural cell behaviors in synthetic cells. So we want to make synthetic cells to behave like a natural cell, but using controllable and very well understood, trackable, engineerable systems. So here's the first one of them. So what we want to do is we want to use synthetic cells to export proteins from the inside of a synthetic cell to the outside, and vice versa, from the outside to the inside. So we engineered this system based on cell-penetrating peptide tags that allows us to make a protein on one side of a synthetic cell membrane, be it inside or outside, and then transport it across the membrane. So first, to just show you that it works, and it works pretty well, this experiment was based on a synthetic cell with a red dye in a membrane. So what we have here is a liposome, phospholipid liposomal membrane that's dyed with a red fluorescent dye. And inside that membrane, we make a green fluorescent protein with a cell-penetrating peptide tag attached to it. After the experiment, that sample is purified on a size exclusion column. So it's just a gravitational column on a Sepharose beads where we divide particles based on size, roughly based on size. And what we see on this chromatogram is a peak in a red channel that shows where is the membrane. So this is where the synthetic cells are. This red peak shows you in those fractions, there is the bulk of the synthetic cell membrane, so the intact synthetic cells. Now, the same chromatogram viewed in a green fluorescent channel shows you where the green fluorescent protein is. And that demonstrates that our cell-penetrating peptide tag export works. So let's look at the right side graph. We started with a membrane. Inside that membrane, we make GFP. Without cell-penetrating peptide tag, after the incubation time, all of the green fluorescent protein is in the same fraction as the membrane. So that means the green fluorescent protein is still encapsulated inside the synthetic cells. But with the CPP tag, with the cell-penetrating peptide tag, most of the green fluorescent protein that was made inside is actually outside. This is the free dye, free solute fraction. So that shows that in the presence of the cell-penetrating peptide tag, the green fluorescent protein diffuses outside of the synthetic cell and elutes from our size exclusion column with the free solute fraction. So basically, the green fluorescent protein was made inside the synthetic cell, and it was exported, or rather passively diffused to the outside, outside environment. So this is all cool, but what can we do with it? So first we asked, can this system be used to drag a payload across the membrane? So we design a system where a cell-penetrating peptide tag is attached to a MS2 RNA binding domain. So MS2 is a viral coat protein of a virus that binds to a very specific RNA sequence, and it binds with a really high fidelity. So we have this MS2 coat protein that's engineered to have the cell-penetrating peptide tag on it, and it binds an RNA payload. In our case, the RNA payload is a fluorescent RNA aptamer called broccoli. So we have this fluorescent RNA that's inside the synthetic cell, and then we have this cell-penetrating peptide tag labeled RNA binding protein that's made inside a synthetic cell. So only if that cell-penetrating peptide tag labeled MS2 binding protein binds to the RNA and drags the RNA across the membrane, only then the RNA can be found outside of a synthetic cell, because RNA will not leak out of the synthetic cell on its own. And if you look at the percentage of the inside versus outside of the labeled RNA, the more of the more protein we make. So basically, the more of the cell-penetrating peptide tag protein we make, the more RNA can cross the membrane. So that shows we can drag a payload across a synthetic cell membrane. So this was one population of synthetic cells importing or exporting RNA payload or protein payload. Now, what can happen if we have two populations of synthetic cells? Here there are two populations. One population expresses T7 RNA polymerase with a cell-penetrating peptide tag. The other population has a plasmid that encodes green fluorescent protein under the T7 RNA polymerase promoter, but it has no T7 RNA polymerase. So that circuit of that resulting expression of GFP can only be closed if the T7 RNA polymerase from the first population, this population called population T7, somehow makes its way to the population GFP. And so what happens is the T7 RNA polymerase is expressed in the population T7, and because it's labeled with the cell-penetrating peptide tag, the T7 RNA polymerase diffuses out of the population T7 and then diffuses into the population GFP. And we can see that by measuring the green fluorescent protein expression from that whole system, the mixed system of those two populations. And if we mix the population T7 and GFP in equal molar ratios, we see some significant above background GFP expression. If we double the concentration of the population T7 by keeping the population GFP constant, we see basically double the expression. If we double the population GFP, keeping the T7 population constant, we don't see as much increase of expression. That indicates that the T7 RNA polymerase is the limiting factor here, which kind of makes sense when you think about it. What has to happen for this experiment to work is that the T7 RNA polymerase has to be made in enough quantity inside the population T7 to saturate the outside environment and makes its way towards the population GFP. So the T7 will always be the limiting factor here. So that's why doubling the amount of population T7 significantly increases our expression from the population GFP, while doubling the amount of GFP carrying population does not significantly increase expression. All of the proper controls, in the absence of T7, we see basically background fluorescence, and in the absence of GFP plasmid, we also see background fluorescence. So what this experiment demonstrated is that you can have cross-population communication between different populations of synthetic cells using a cell-penetrating peptide tag labeled active enzyme proteins. We also demonstrated that this cell-penetrating peptide tag system works with other enzymatically active proteins, for example, luciferases. And basically what this system demonstrates is that you can make something in one liposome compartment, in this case, inside a synthetic cell or on the outside of a synthetic cell, and the protein product can be exported across that membrane, so it can be dragged across that membrane. We also can drag functional payloads, so functional nucleic acids. We also tried, and I know that that's a question I often get, so I might as well answer it right now. We tried dragging mRNA payload. Unfortunately, we never got the mRNA payload to work. We have a really good luck dragging smaller payloads like the broccoli aptamer that I showed you. We tried many times in different configurations to drag a functional mRNA, basically gene coding mRNA as a payload, and we're never able to see significant detectable transport. So that we're assuming that the mRNA is just too big to drag as a payload across the membrane using this system. But smaller functional RNA can be dragged. And then we also show that proteins can migrate from one population of synthetic cells to another, and then activate genetic circuits. And some of the cool applications of this technology can range from very practical. You can use synthetic cells as those little liposomal bioreactors circulating in a bloodstream. As we know, a couple years ago already, Avi Schroeder demonstrated that synthetic cells can be used to shrink tumors in live mice. So there's promise that synthetic cells can be used for biomedical applications in mammals. And this can also be used for metabolic engineering, where we exchange enzymes between different populations. We can also imagine further building up synthetic cell systems, where we build nested liposome systems that imitate organelles, where we can build encapsulation within encapsulation. And we can also study communication between synthetic cells. We can build systems where synthetic cells exchange signals, molecular signals or protein signals. So this is one project that, one kind of a liposome-based project that we did in order to build in a natural cell functionality into a synthetic cell. Another project kind of inspired by that two population of liposome system, where we were exchanging a simple T7 RNA polymerase, is a project on engineering antibiotic resistance in synthetic cells. So what we have here is two different populations of synthetic cells again. And again, they're liposomal bioreactors with TXDL bacterial TXDL encapsulated inside. So we have two populations of liposomes, those synthetic cell liposomes. They both have GFP inside them. They both have a alpha-chemolysin, which is a membrane protein. It's a protein that spontaneously forms a membrane pore. And one of those populations has a kanamycin resistance gene. So the alpha-chemolysin is needed because kanamycin will not cross the membrane on its own. So only in presence of alpha-chemolysin, we can uptake the kanamycin into the synthetic cells. So now we're adding kanamycin, which is a ribosomal disruption antibiotic. And if you look at those two populations under the microscope, the population that has the kanamycin resistance keeps making GFP. So GFP here is our proxy for a synthetic cell metabolism. So in that population with kanamycin resistance, we still can see GFP. In the population without kanamycin resistance, we don't see any GFP production. And now the labels of those synthetic cells again were, the membranes of those synthetic cells were again labeled with red fluorescent dye, and it shows us that the membranes remain intact. So in the population without kanamycin resistance, the kanamycin doesn't do any mysterious thing of disrupting the membrane itself. It just disrupts the metabolism. It just disrupts the GFP production inside the synthetic cell. So now looking at it quantitatively, in the population without resistance, as we increase the concentration of kanamycin in a sample, the GFP production significantly decreases all the way down to no detectable production at 2.5 micrograms per milliliter of kanamycin. In the population with resistance, the GFP production decreases some. We attribute that to leakage of the kanamycin inside some of the more permeable membranes, but the production remains. We still detect the GFP metabolism all the way down to the highest tested kanamycin concentration. So now let's take two different populations of synthetic cells. One of them has alpha-chemolysin channels in a membrane. So that's this population with alpha-chemolysin and M-cherry fluorescent protein. So that's a red fluorescent protein. The other population has no alpha-chemolysin channels, but it has GFP fluorescent protein. We mix those two populations in one test tube. And in the absence of kanamycin, under the microscope, we can observe both of those populations expressing their respective fluorescent proteins. If we add kanamycin to the system, we only see the GFP producing population. And that's because the GFP is the one that doesn't have the membrane channels. The one with alpha chemolysin that is supposed to express mCherry is not expressing the red fluorescent protein in the presence of kanamycin. And again, we can quantify that. GFP expression does not significantly change as we increase the kanamycin concentration, but the mCherry expression significantly goes down to undetectable levels as we increase kanamycin concentration. So that demonstrates that the membrane channels are indeed necessary to get the kanamycin into the cells. And then we can do the opposite experiment where the channels are present in the green fluorescent protein synthetic cell population, and the red one is the one without channels. So in presence of kanamycin, we only see red cells under the microscope. And again, looking at the quantitative fluorescence from both channels, the red fluorescent protein is made at increasing concentrations of kanamycin, but GFP concentration, GFP production goes down significantly as we increase kanamycin concentration. So membrane channels are significantly are necessary for uptake of the kanamycin to the synthetic cells, and the antibiotic resistance protects synthetic cells from kanamycin. So now that's kind of flashback to yesterday's talk. Those of you who have seen Neha's talk on DNA-mediated liposome fusion will remember that we have this system where we can fuse two or more populations of synthetic cells using DNA tags. Those complementary DNA tags are essentially molecular zippers. They allow us to fuse populations of synthetic cells on the same principle as the snare proteins, the proteins that fuse our synaptic proteins to the membrane of the synapse. And so we have the synthetic cell populations labeled with those complementary DNA tags, and when the DNA tags hybridize, the synthetic cells fuse. So what we did here is we have two populations of synthetic cells, one of them with a green fluorescent protein, the other one with alpha chemolysin. We fused them. So now we have this daughter population that contains both alpha chemolysin and green fluorescent protein. And we add kanamycin to the system. So we, in the absence of the fusion, so in the samples where we did not perform the fusion experiment, so the DNA tags were not complementary to allow fusion, the GFP concentration GFP production does not decrease with increased kanamycin. That is as to be expected because the GFP population itself does not have alpha chemolysin. In the fusion experiments where we did fuse or mate those synthetic cell populations, the GFP concentration significantly decreases with the increased kanamycin concentration. The reason it does not decrease all the way down to zero, as we've seen in other experiments, is because the fusion system is not 100% efficient. So there's some GFP population members that did not fuse with the alpha chemolysin members. And so that provides that residual GFP, that still detectable GFP production at higher kanamycin concentrations. So in this case, we demonstrate that this fusion or a mating system is harmful to cells because the GFP population was doing perfectly fine on its own. But when it fused with this alpha chemolysin population, it started to uptake the toxin. Now let's imagine the opposite system, where we have, again, two populations of synthetic cells. One of them contains the GFP plasmid and an alpha chemolysin plasmid. So this is the population that has membrane channels and a GFP plasmid. The other population has a kanamycin resistance gene, but no channels. We fuse those populations, and now we have a daughter population that has channels and kanamycin resistance. If we look at GFP expression in the absence of fusion, it goes down to undetectable levels with increased amount of kanamycin, because the GFP population has channels, so can uptake kanamycin. Now, in the presence of fusion, the GFP expression remains detectable even at the highest kanamycin concentration. And that demonstrates that the fusion is actually beneficial to cells because the fusion introduces the kanamycin resistance gene to the population, and allows us to preserve some of the GFP expression in the presence of kanamycin. So this system demonstrates that we can have antibiotic resistance models in synthetic cells, and we can model spread of that antibiotic resistance through the population of synthetic cells. So we can have antibiotic resistance that spreads between the populations of synthetic cells, antibiotic resistance that's beneficial to the populations of synthetic cells. We can use that for the obvious applications of modeling arising of antibiotic resistance, which has kind of practical applications, but we can also use it as a model system to model spread of different traits, different evolutionary advantageous or disadvantageous traits in the population. So it's basically controllable horizontal gene transfer system, where we can see how different traits can spread through different populations. So these were two liposomal synthetic cell projects that I wanted to show you, to kind of show you the capacities of the synthetic cell systems, liposomal synthetic cell systems. Now, for the last few minutes, I would like to move away from the liposomes and show you one practical metabolic engineering project in our lab that shows kind of a practical side of the kind of extension of the synthetic cell engineering that we're doing. So there's no liposomes in this next project. It's just cell-free protein expression system. And it's a cell-free protein expression system that's lyophilized on paper. So Jim Collins demonstrated a few years ago that you can take a bacterial TXTL and lyophilize it on a piece of filter paper. And now what we demonstrated is that you can split that system. So you can basically take a genetic circuit in cell-free system, split it into a few different elements, lyophilize each of those elements on a separate piece of a filter paper, and then put them together into a sandwich that reconstitutes the expression. And so what we see here is we have this three-part system where a green fluorescent protein plasmid, the T7 RNA polymerase, and the translation machinery are all brought in on a separate pieces of filter paper, put together into a sandwich, and the circuit is reconstituted. We can also isolate the protein product from the supernatant above the reaction. What is it good for? Well, it's good for metabolic engineering. The idea here is that we can build a complex metabolic engineering systems, for example, a system that produces different kinds of small molecules from different genes and isolated on separate filter papers. And the idea here is that allows us to build metabolic engineering systems on demand. And the reason we're doing it is because we would like to be able to assemble different metabolic engineering pathways on short notice on demand. And why do we want to do it? Well, for production of pharmaceuticals or bioactive compounds or nutrients on demand in small quantities. And the reason we're investigating it right now is because we want to use it as a biomanufacturing platform for long-term space missions. And so recently we demonstrated that this concept of astropharmacy, so this concept of using cell-free systems and even whole synthetic cells for production of small molecules on demand, has legs. It can be proposed to use under a spaceflight conditions. So I was very lucky to be part of this amazing team that set out to test if a cell-free reactions, the proxy for a synthetic cell metabolism, can function in the spaceflight condition environment. And so what we demonstrated is we had two systems, an functional RNA aptamer and a small protein made in cell-free system. We loaded it up on a CubeSat rocket. We launched it. That was the first time my own payload launched up to space. So I'm showing everyone that video because I had to be up at 3 a.m. to watch that video. So I'm obviously super excited about it. It launched, it didn't blow up the rocket. It came back, that was a sample return mission. We analyzed it, and now we know that it's possible to use cell-free system under the spaceflight conditions. And so that basically demonstrates that it's possible to have a cell-free system that works under spaceflight conditions, and now we're setting out to try more complex systems to see what are the limits of this. Can we use it for practical applications? And I would like to conclude the research part of my talk. I would like to thank everyone in my lab. I would like to thank all the members of my lab. I would like to thank all of the funding agencies that made the work I demonstrated here possible. And I would like to take 2 more minutes to introduce you to this BuildACell initiative. So BuildACell is a international open collaboration that is focused on advancing engineering of synthetic cells. And by advancing, we understand advancing it on every possible level. So doing science outreach, doing communication with the public, doing communication with other scientific disciplines, collaborations with scientific disciplines, but also lobbying with our government, convincing our government that this is the field that's worth investing into, but also that this is the field that needs current regulations. We're working on biosafety, biosecurity implications of synthetic cell engineering. We're working on making this field safe, making this field inclusive. So if you're interested, please join us. Please join any of the activities we're doing. We have weekly seminar series. We have working groups. If you have any interest in contributing, I would really like everyone who's interested in this field to join. And the kind of guiding principle behind establishing BuildACell is to share everything, is to be able to integrate our field, is the kind of conclusion that no single country, no single lab, no single nation is capable of doing this amazing, huge job of engineering a cell from scratch. We need to integrate across our different national systems. We have, our own funding system here in the US, Europe has their own systems, Asia has their own systems. We need to be able to work together, share data. We need to be able to share also unpublished data, share ideas, and work as a single community. And the goal of it is that we want to be able to speak in one voice to command large resources that this field deserves. And as an example, we're using other fields of science. Now, astronomy or physics were able to unite and command those large billion-dollar resources to build shared infrastructure like radio telescopes or particle colliders. If our field, the field of engineering life from scratch, was able to unify, if the synthetic biology was able to speak in one voice, what would we ask for? I don't have an answer to that, but I would like to make a case for the point that building life is, on one hand, this amazing basic research endeavor, and on the other hand, it has tremendous practical applications. So it's not unlike other areas of science that we're able to unite and command large resources. So we should do that too. And that's one of the things that the BuildACell initiative is working on. And I would like to ask everyone who is interested in this field to please join us. Please join the BuildACell community. Please work with us. And I see I'm out of time, so I will stop here. And I want to thank again everyone, thank the organizers, and I will take questions. Thank you very much.
Thank you, Professor Adamala, for a great and exciting talk. I'm really impressed to see that it's super nice to see this collective behavior of your kind of synthetic cells, the transfer of information between them. It's something exciting. So we have a time for a few questions because we are kind of limited in time. So there is a first question: why does a lysosomal synthetic cell without alpha hemolysin has decreasing reported protein expressions with increasing concentration of antibiotic?
Why does a lysosomal synthetic cell without we attribute that to certain degree of leakage of kanamycin into the synthetic cells. The kanamycin is supposed to be impermeable to cells, but we know that at higher concentrations, it's possible to have some leakage. And kanamycin is a very high affinity antibiotic, so just few molecules would be enough to shut down ribosomes. So that's what we attribute it to, is there is a little bit of leakage through our synthetic cells.
Okay, there is a question from Timay. Thanks a lot for your interesting talk. I have a question. Does your liposomes with protein channel for loading RNA into the liposomes? It without channel? Is it if without channel, is it possible? How to increase the loading of RNA into liposomes?
Oh, so we're not using channels to load RNA into liposomes. The RNA crosses. The crossing of RNA across the membrane is mediated by a cell-penetrating peptide tag protein, and that protein we're using, the penetrated, does not create channels. It translocates across the membrane, so there's no channel in this case. And to increase the loading, we basically have to increase the concentration of the protein, since the loading is concentration gradient based, so it's completely passive. The more you have on one side, the more is going to cross. So to push the loading, you just have to increase the concentration on one side.
Okay, thank you. And maybe the last question, because we are over the time already. Hey, Kate, great presentation. Is there an evolutionary biology equivalent to a Turing test for all, or test in which you can identify the point at which synthetic cell might begin optimizing autonomously beyond what is designed in?
That is a great question. I believe there is an AI solution for that. I don't have it right now. I don't know at which point the system will take over with the Darwinian evolution. We think we're still missing few of the hardware elements. So for example, we need a self-replicating genome and just physical self-replication. But I think there definitely a point like that exists, and it would be really cool to get some machine learning on it, able to identify a level of complexity that allows that kind of a runaway evolution system.
Thank you very much, Professor Adamala, for this really nice talk.
Thank you.
There were more questions that you could answer, as unfortunately we are running out of time. I don't know, maybe you can see these questions and answer them afterwards if it's possible. Most probably it is. But now I would like to move to the next speaker, if it's possible. So thank you very much, Professor Adamala, again.
Thank you, and thanks everyone. If anyone has questions, you can find me by email.
Thank you. So our next contributed talk will be delivered by Michael Levin. From Dr. Levin obtained his PhD in physics from the Université Denis Diderot from Paris in 2011. And then he continued with his research career at UCLA as a postdoctoral fellow in the group of Professor Dolores Bozovic. And since 2015, Dr. Michael Levy is a postdoctoral fellow in the group of Professor Roy Bar-Ziv in the Department of Chemical and Biological Physics at Weizmann Institute of Science, Rehovot, Israel. So today, Dr. Levy will speak about cell-free autonomous biogenesis of ribosomal subunit. So shalom, Michael, and batzlacha.
Thank you. Thank you very much. So let me share the screen. Okay. So thank you very much for this nice introduction. So I want to present you the work that I did during my postdoc at the Weizmann Institute. And it's going to be about ribosomes. So in this big goal of making a synthetic cell, we focused on making a ribosome. The ribosome is the machine that is actually making in a real cell all the elements. So it's essential for growth, it's essential for self-replication, it's essential to make all the enzymes. So a self-replicating artificial cell will require ribosomes. Again, to emphasize the importance of ribosomes in a real cell, in E. coli, for example, 10 to 20% of proteins are ribosomal proteins, ribosomal proteins. 85% of the RNA is ribosomal RNA in E. coli. So it's an essential machine, if you will. So here I show an image of a ribosome. You see it's made of two parts. The small subunit here in blue, made of 21 ribosomal proteins and one long ribosomal RNA. So the long RNA is this blue spaghetti. And you have a large subunit, the red one, that is even bigger. So now you see the problem. Okay, the problem is that it's maybe the most complex machine that is required in artificial cells that we need to make outside of a cell. So that's the question. Can we reproduce ribosome biogenesis in a synthetic environment, not in a cell? The cell knows how to do that. Can we do that in a synthetic environment? So it's too much for us, actually. So we decided to focus only on the small subunit. So this talk will be only about making the small subunit in a synthetic environment, not the large subunit. So what's the objective? The objective is to reproduce ribosome biogenesis outside living cells. We want to start with synthetic genes, coding for all the elements of the small subunit, put that in a cell-free gene expression reaction with purified components. We know the composition precisely of this gene expression reaction. And we want that by putting the genes and the gene expression reaction together autonomously, we make the elements and they assemble together in a new small subunit. We want this to be an autonomous process. So now you see the problem. The cell-free gene expression reaction is limited. You can make a limited amount of material with that. So you will end up with all the elements made from the DNA, but in a dilute environment. And what you need to build a new small subunit, you need 20 binding events on the same object. So what's the chance to have that in a dilute environment? It's nothing. Okay, the probability to have all these binding events assemble the small subunit in a dilute environment is nothing. So you need a trick. You need a way to do it experimentally. So our method was the following. Instead of having the genes diluted everywhere in the volume in a reaction in test tube, we concentrate the DNA in a DNA brush. You can put in this DNA brush on the surface 1,000 DNA molecules with a single gene on them, 1,000 DNA molecules per micrometer square. So you end up in the DNA brush with a DNA concentration that is comparable to the bacterial nucleoid, basically. And now the idea is that if you concentrate the DNA on the brush, hopefully you will concentrate all the gene products, all the proteins and the RNA around this DNA brush. So you will reach high local concentration of everything, high enough to drive assembly of the small subunit. So you put all the genes coding for the small subunit together, and hopefully they will assemble. That's the first trick. The second trick is you trap back the assembly complex on the surface, close to the brush. Why? Because at the beginning, it's not complete. The assembly complex is not complete. It's not a full small subunit. And then it's going to diffuse away far from the high local concentration. But if you trap it close to the source, it will be completed because it's now close to the source of material, of all the proteins. So these are the two tricks that we used. How do you trap the assembly complex on the surface? Well, you modify one of the proteins with an HA tag. It's a small peptide that will bind to an antibody on the surface. And you can put this tag on any of the proteins. So we will play this game. We will change the place of this HA tag to trap on the surface all the proteins, the different proteins. And the RNA in almost all the talk, the RNA will be the fluorescent object that we will look at. So the trapping of the RNA on the protein will lead to the trapping of the RNA to the surface, and that will be the signal. So now I need to prove that the brush, the DNA brush, is a source of material, that it produces this high local concentration. So we need to study gene expression on a DNA brush. So this is a DNA brush, dimensions about 100 micrometers in this case. To study gene expression on a DNA brush, we use turf microscopy. Turf microscopy is a fluorescent microscopy technique that illuminates, that excites only the first 100 nanometers above the surface. And that's the height of a DNA brush. So it's a very good technique to look at what's going on in the DNA brush. So now what we're going to do, we are going to do different experiments, each time labeling another actor of gene expression. So we label, so follow the green, we label the RNA polymerase here in green, and we start gene expression on a DNA brush. We see localization of the RNA polymerase on the brush. No big surprise, the polymerase binds to the DNA directly. Now we do another experiment. RNA polymerase is not labeled anymore, but we make an RNA that is now fluorescent. The RNA that you make is fluorescent now. And you see that it also localized on the DNA brush. Now you label the ribosome. Only the ribosome is now labeled in green. And you see that the ribosome localized on the DNA brush. That's more surprising because ribosomes, they bind to the RNA, not to the DNA. So localization of the ribosome on the DNA brush was not trivial. And actually it was even less trivial that if you do it on a DNA brush that is not coding for a gene, that is passive, non-coding DNA, ribosomes are excluded from the brush because the brush is so dense, ribosomes, they don't want to get in. They are excluded from the brush. So you really have some kind of a non-equilibrium system where activity drives in ribosomes inside of the brush. And last, and that will be the proof that the brush is a source of proteins, if now nobody is labeled, but you make a fluorescent protein, the fluorescent protein that you make will localize also on the brush. So like that, we have a good indication, if you want, that the brush will act like a source of product, so will probably drive high local concentration of everything. Okay, so now let's look at the assembly of the small ribosomal subunit. You need to know a few things before we look at experiments. The thing that you need to know are the following. Imagine that the ribosomal RNA is this green line. All the proteins binds directly to the ribosomal RNA. Okay, the ribosomal RNA is the core of the small subunit. All the proteins, they will bind to that. But some proteins called the primary proteins, P, they bind directly to the ribosomal RNA no matter what. Some other proteins called the secondary ribosomal proteins, S, they bind also directly to the ribosomal RNA only if some of the primaries are already on the ribosomal RNA. So for example, 16 will bind if 4 and 20 are there. That's the meaning of the arrows. And last, some proteins will bind, they are called the tertiary binders, will bind to the ribosomal RNA only if some of the secondary and some of the primaries are already there. So you have some kind of a binding dependencies of the proteins on the RNA. So it's a well-organized system, and we are going to use that. We are going to label the ribosomal RNA. That will be the fluorescent object, and we'll put the HA tag on the different proteins. So all the proteins will independently bind to the surface in different cases. And we will study in what condition does the RNA bind to the surface through this specific protein. And that will be the signal because we will look at it in turf microscopy. So we have a signal only if the fluorescent RNA binds to the protein that is on the surface. The first experiment that we want to do is very simple. We test all the two bodies. We mix in the DNA brushes the RNA fluorescent and a protein with the HA tag. Each time a different protein with the HA tag. And you can scan the 20 possibilities on a single chip. It's a cheap approach. When you have a signal, it looks like that. This is the DNA, the 3 spots. The signal propagates from the spot, and they look like this. This is the hexagonal pattern because we have a way, it's an experimental detail, but it's interesting. We have a way to localize antibodies on these hexagons. Like that, we have a specific signal. It's not a non-specific binding. It's something that is related to antibodies. Antibodies are localized on these hexagons. So if you see a signal on these hexagons, it means that first, the protein HA bound to these antibodies on this hexagon, and then the fluorescent RNA bound to the protein. You do it for all the proteins, and you see that you have a signal only for the primary proteins. Only primary proteins will bind the RNA, not the secondary, not the tertiary. That makes perfect sense. The secondary would need other proteins in order for the RNA to bind. So just to make it a little bit more visual, I put the data on the map. And you see that in this case of a two-body, you have only the first line basically that lights up, meaning that you have this specific interaction with the primaries and no secondary, no tertiaries. So now you jump, okay? We looked at the two-body. Now we do the same thing, but we add all the other proteins around. So now you will still scan all the proteins, the different proteins with an HA tag, but all around you have the rest. So now all the proteins HA have everything that is needed to bind the RNA. All the other proteins are there. And when you do that, you see that you have a signal, not just for the primaries, and actually some signals are more stable in this case, but you also build a signal for secondary and tertiary proteins. So you really see mediated interactions between the protein and the RNA, thanks to other proteins. And again, if you look at it on the map, now you see that it's not just the first line that, not only the primary that lights up, also the secondaries and also the tertiaries. And the interesting thing is that they don't light up at the same time. Some lights up first and others lights up later on. If you look at the beginning of the signal, you take the beginning of the signal and you plot it together, you can find out the timeline of ribosomal subunit assembly. In this case, you see the first binders and the last binders. And you see that basically in about an hour, you build up the small subunit. So interestingly, in a cell, it takes 3 minutes. So there is something that is different in an actual cell. Next, I want to show very quickly, I'm not going to go into the details, that this approach of a chip where you can put and remove genes is very useful to work with systematic deletions and build this map. Find out this map back. Let's take an example. If we look at the binding of, let's pick 6, this protein 6, binding of 6 to the RNA. What are the conditions for 6 to bind to the RNA? This protein number 6 to bind to the RNA. So 6 will be the protein with the HA. Now you put together in presence of 6 all the relevant proteins. So gray square means protein present. The signal here, that is the signal that I showed you, that is now shown in a column map, showed that there is a binding of 6 with the RNA. You remove 11, you still have some signal. A bit lower, you still have some signal. You remove 8, so white square means absent. You reduce your signal. You remove 8 and 11, you reduce a bit your signal, but you remove 15 and only 15, you kill your signal. That means that 15 is required for 6 to bind the RNA. You get the idea. You can play with systematic deletions and find out who needs who to bind to bind the RNA. Okay, the last experiment I want to discuss is the following. You put on the brush all the genes to build a small subunit. But now, no protein has the HA tag. Now, what is on the antibody is large subunits that are purified from bacteria, from E. coli. You want to see if the newly made small subunit that is now fluorescent can bind to the large subunit that is on the surface. If you put all the proteins, you have a signal. The small subunit binds to the large subunit. If you remove only 3 proteins, 13, 14, 19, that are known to help the interaction between, to be involved in the interaction between the small and the large subunit, you reduce your signal. You remove all the primary proteins, You reduce your signal and you remove all the proteins, you kill your signal. So it's a very specific signal, meaning that the newly made small subunit binds to the large subunit that is on the surface. Okay, as a conclusion, we recreated the biogenesis of E. coli small ribosomal subunit by synthesizing and capturing all the ribosomal proteins and RNA on the chip. That was the technique. Surface confinement provided favorable conditions for autonomous stepwise assembly of the new subunit. And interestingly, the new subunit is now separated from the new one is separated from the old one that is used. You need to put old ribosomes to build new ribosomes, but now the new ones are on the surface, the old ones are in the volume. You can separate old from new. Real-time fluorescence measurements revealed hierarchical assembly, cooperative interactions, and stable intermediates, and specific binding to large ribosomal subunit. That was this last experiment. Now, using only synthetic genes, this methodology is a step towards creation of a self-replicating artificial cell. It's also a general strategy for the mechanistic investigation of many multi-component macromolecular machines, not only the ribosome. So with that, I would like to thank the Barziv lab, actually, especially Rui Barziv, the professor, and Shirley Dobe, who was essential for this project. And I would like to thank you for your attention. I'm open for any question.
Thank you, Dr. Levy, for a very interesting talk. We have actually 2 questions currently. The first question, will the product from DNA brush interact or even mix with the ribosome in expression system?
Again?
Will the product from the DNA brush interact or even mix with the ribosome in expression system?
It's a possibility. It's a possibility. We are now trying to mix species. Now it was E. coli with E. coli. We were using E. coli cell-free expression system, called namely the pure system, with E. coli genes to make the E. coli small ribosomal subunit. So you may have interaction between the two. Now we are looking at E. coli ribosomes making other type of ribosome from other species. And in that case, I mean, you may have some, I would say that mixing, which would be very interesting actually, but it's less probable, but it's a possibility.
Okay, thank you. The next question, would it be possible to play with the stoichiometry of the ribosomal protein genes on the chip to optimize the assembly?
Yeah, we did it actually. In our case, the most important factor was the ratio between the RNA and the protein with the HA tag for technical reasons. But there is definitely room for optimization in this regard.
Okay, and maybe then the last question from Ivan Ivanov. Excellent talk. Did you manage to prove functionality of the fully assembled ribosomes beyond binding?
No. The closest we could get from functionality was the binding with the large subunit. To get to activity, it would require a major change of the platform, because you have this mixing between old ribosomes that are here to build the new ribosome and the new ribosome. So it's not trivial to find a way to separate activity from only the new ones. We are thinking about it.
Okay, thank you very much, Michael. Again,
thank you.
So now it's a good timing to move. Good time to move to the last, not last, our second feature speaker for today. and he's a James Carother.
Mm-hmm.
James Carother is the Dan Evans Career Development Associate Professor of Chemical Engineering and Adjunct Associate Professor of Bioengineering and member of Molecular Engineering and Science Institute and Center for Synthetic Biology at the University of Washington. Previously, James was a postdoctoral fellow and research scientist with J. Kisling at UC Berkeley. And before that, he was a graduate student at Harvard, where he earned a PhD with Jack Zotzak. James has a bachelor in molecular biophysics and biochemistry from Yale. He has received the University of Washington Presidential Innovation Award and the Alfred Sloan Research Fellowship. Today, Professor Carother will speak about synthetic cell system for scalable bioproduction of plant natural products. I'm looking forward for your talk.
Thanks very much. Are you able to see my slide?
Yeah.
First slide. Okay, great. All right. Well, thanks so much. I appreciate the chance to talk to you all today about some of the work that we've been doing. But first, I want to give you a little context or talk a little bit about what our group does. So our group at the University of Washington essentially does three things. We use large-scale computational simulations to design metabolic control systems. We engineer CRISPR-Cas transcriptional networks to implement those designs, and we build aptamer biosensors as ways of reading out the products of the systems that we build, and also working our way toward dynamic regulatory control systems. So we've organized our work around applications in bioproduction using microbes and cell-free systems. Specifically with our collaborators, we're developing cell-free and synthetic cell systems for a wide range of applications, including for plant-derived drug biosynthesis, antibody diagnostics, information processing, and as bioproduction systems to convert carbon dioxide into industrially important chemicals. Now, there, of course, have been a number of successes in recent years, taking steps toward bottom-up construction of synthetic cell-like systems that use cell-free expression systems to recapitulate, but in many ways reimagine how cell-like behaviors can be constructed. So we've seen a lot of really nice examples in this conference of people developing synthetic cell systems that employ different boundary systems that utilize distinct metabolic substrates and activators, and then have different chemical information systems to program functional outputs. So this is all terrific. And of course, there are a number of efforts underway to develop cell-free expression systems as platforms for the bioproduction of medically and industrially important compounds and materials. Nonetheless, efforts toward developing genetic control systems that are suitable for programming complex functions in cell-free systems remains at an early stage. And I think there are a number of reasons for this, but it's been difficult to integrate multiple components. The strategies have not generally been scalable, and the responses produced by these systems are typically triggered, but not dynamic. So our most recent efforts have focused on engineering versatile CRISPR-based gene regulatory networks that are capable of programming dynamic functions in cell-free systems. And the tools that we've developed enable us to rationally tune target gene expression dynamics and give us a framework for building scalable CRISPR AI networks as regulatory programs and cell-free expression systems. And so we're now applying these to problems of coordinating the timing and expression levels of lots of genes in engineered metabolic pathways, including plant natural product biosynthesis. So now let me give you an outline of what we're going to talk about today. So first, just to kind of set the baseline here. So the creation of CRISPR-based tools for programming gene expression, I think it really has there's an opportunity here to accelerate the development of bio-based production systems in lots of different platforms, including microbes, but also cell-free systems. The application of CRISPR tools for transcriptional repression or CRISPR inhibition in bacterial metabolic engineering in particular is well established. By comparison, the creation of CRISPR tools for transcriptional activation in bacteria has lagged. We've been working now for a number of years with our collaborator and my good friend Jesse Zalatan at the University of Washington to develop new tools for bacterial CRISPR activation. So what we've done is identified effective transcriptional activators. We've systematically defined rules for effective bacterial CRISPR activation, and we're creating approaches for encoding functions in engineered guide RNAs. And so we've shown that these CRISPR tools can drive expression of multi-gene biosynthetic pathways from multiple promoters and multiple hosts, including a couple of different bacteria and also E. coli-derived cell-free transcription translation systems. So what I'm going to do in this presentation is describe our most recent results and outline some strategies for integrating CRISPR activation with CRISPR inhibition to create new capabilities for building dynamic genetic control systems in synthetic cell systems and for developing cell-free systems as programmable platforms for bioproduction. Now, I want to begin just by giving a little context here, why it is that we care about these bioproduction problems. So in principle, there's a vast space of medically and industrially important chemicals and materials that could be produced using bio-based systems. In fact, it's not always appreciated that bio-based chemical production is already a $160 billion industry in the United States, and that we're on track for about 25% of all chemical production to be bio-based by 2025. Of course, there have been a number of notable recent successes in applying synthetic biology and metabolic engineering for applications in bioproduction. But the fact remains that this is still a challenging, time and resource-intensive undertaking. And I think that one of the reasons here, really the fundamental challenge, is that you have to program the activities of a large number of gene products, and they all have to function together as a system inside a cell. This is really a significant challenge, kind of intellectual and also practical challenge, how to develop the theoretical but also experimental frameworks for turning a desired output into a set of programs that you can actually implement and on a reasonable time scale. Now, there are a number of ways of thinking about, really kind of why this is a challenging problem. And I'm going to frame it this way because I think the counterpoint to a naturally occurring biological system and an entirely synthetic biological system, I hope will be clear when I do this. So, one of the main challenges is that if I introduce new multi-gene functions to a cell, then what's really happening is I'm introducing these functions to a pre-existing network. And that network is generally perturbed when I introduce new functionalities. And of course, the network itself is also complex. This is our nutrient stress genes in E. coli, where each of the circles represents an individual gene. And then they're showing the connections between the genes, and the size of the node tells you something about the strength and how many connections are made between genes in the network. Now, and predicting what's going to happen when you introduce new functions to a network like this one, what perturbations will actually arise is not a trivial undertaking. Of course, you might think that simply switching to a cell-free system would alleviate all of these problems, and it certainly does. This is one of the main advantages of a cell-free system, right? You can get rid of a lot of the metabolic network and just recapitulate the functions that we want. And so that's very important, but it isn't actually a kind of get out of jail free card, particularly as the pathways and networks that we begin to develop for cell-free systems increase in complexity. And so, to help kind of explain why this challenge is still there, why we haven't solved it just by moving to a cell-free system, I want to introduce a pathway that we're working on in my lab. So we've organized a lot of our efforts around the production of industrial aromatics, including a molecule like this one, p-aminostyrene, shown in the blue box there. This is a vinyl aromatic whose polymers have tunable optical and mechanical properties that are useful for a bunch of advanced applications. Now, in principle, you can produce p-aminostyrene in about 15 steps from glucose, whether in a microbe or in a cell-free setting. So you could go from glucose to charismate and then add a heterologous or non-native pathway to an organism like E. coli, and you could convert charismate into p-aminostyrene. Now, in practice, when you introduce these new metabolic functions to a cell or to a system, you bring along with it what I like to refer to as control problems. So there are intermediates here that are readily diverted. The flux is readily lost from the pathway. There are other enzymes that consume aromatic amino acids, so the precursors both of cell-free gene expression and also the precursors of the pathway itself. There are other intermediates that poison the reaction. So these would either poison gene expression or poison possibly even the catalyst that you're using to do the chemistry in the engineered pathway. So, I'm kind of showing you a schematic or an illustration here for our version of the pathway that we built in E. coli. But the fact is that these are really going to be present essentially in any biochemical system that you can think of, whether they're naturally occurring or wholly synthetic. So these kind of control problems are, there will be different flavors or different versions of these depending on the system that you're trying to build, but these are going to be present irrespective of the location where you're building the system. Okay, so what we've been thinking about is, how can we develop some designable expression tools that we can use to rapidly program the timing and expression levels of large numbers of genes in these systems, whether those systems are microbes or cell-free or something else, kind of a hybrid of the two. So our strategy here, and I'll use this also to explain just kind of the particulars of the mechanisms for doing CRISPR inhibition and activation in bacteria in the systems that we've developed. I should say bacteria and also bacterially derived cell-free systems. So to target genes, we use nuclease defective Cas9, so dCas9, with a guide RNA that's going to specify a target site on the DNA. So if we target this complex to a promoter or an open reading frame, then this will give us gene repression, so CRISPR inhibition. If we want to enable simultaneous activation, then we use modified guide RNAs. We call these scaffold RNAs that have a 3' MS2 hairpin that recruits a transcriptional activator that's fused to the MS2 coat protein. And so we can express multiple guide RNAs and multiple scRNAs to inhibit and activate genes simultaneously. So in this case, sgRNAs targeted to promoter or an open reading frame will give you CRISPR-I. scRNAs targeted to an appropriate site upstream of a promoter will give us CRISPR activation. And it's this combination of repression and activation that we want to use to implement transcriptional programs. All right, so now what I'm going to do is tell you or show you that we're developing some new rules for improving and kind of generalizing synthetic gene regulation in bacterial systems. So this will get us started. So we started this project several years ago, and the first thing that we had to do was identify some candidate transcriptional activators that we could append to that we could recruit using our modified guide RNAs to give us CRISPR activation-directed transcriptional activation in bacteria. And so the rest of the work that I'm going to show you use this system as the basis for building these kind of fully programmable platforms to activate some genes or to target some genes for activation and other genes for inhibition. And so I'll begin with some of our results, kind of investigating some questions that came out of that earlier work. So which promoters can we actually activate? What's the impact of the promoter sequence or the presence of transcriptional repressors or other rules about the organization of the promoter region as far as our ability to activate them? Could we get a clearer view of what we'd noticed, some kind of CRISPR activation distance dependence on the amount of activation that we could get relative to the transcription start site? What we really wanted to do was understand how to most effectively activate genes from engineered promoters and develop a set of rules that would allow us to activate genes within bacterial genomes. Now, of course, that last piece is obviously of no concern really for application of rules for cell-free engineering, but nonetheless, that was an important part of the work that we were doing there. So I'll just give you a couple of examples of the kinds of questions that we investigated. So the first one we wondered about was whether our new system, whether the intrinsic promoter strength would affect the level of CRISPR activation that we could achieve. And so for these experiments, we're using a test set of minimal promoters that we've designed that span a 200-fold range in basal expression levels. So from barely detectable to kind of maximum expression, which incidentally, if this means anything to you, this would be something like strong tetracycline-induced expression in an E. coli strain. And so, what we found is that CRISPR activation is sensitive to the promoter strength, which is not necessarily surprising. You can't significantly activate genes with high levels of basal expression. So if they're already turned really on, you can't activate them more. And at the other end of the spectrum, we weren't able to significantly activate expression from promoters with the weakest levels of basal expression either. But then there's this kind of broad range in the middle, the Goldilocks space there, where you can significantly activate the amount of expression that you get from the system. Okay, So in our original paper, we'd seen this kind of strong dependence on the distance between the transcription start site and the guide RNA target site within the promoter region. So now what we're doing is adding more resolution to that analysis. And when we did that, we found something interesting. It's a very well-ordered kind of rise and then fall and then rise again in the amount of activation that you get as you move away from the transcription start site. So that's going from the right-hand side, the +1 site, and then kind of marching your way back to the left at -81 is kind of the maximum peak of activity. And then you kind of see these peaks and troughs. And presumably what's happening here is the CRISPR-A complex is rotating around the DNA. It needs to be on the same side of the DNA as the polymerase, giving rise to these peaks of activity. And so it's through these experiments and a number of other ones that I don't have time to kind of present today, but this kind of gave us a picture of the complexity of the task of activating promoters using CRISPR activation. But nonetheless, it was also clear to us that there are routes for using these rules to design new CRISPR-A components that we could hopefully use to build multi-gene expression programs. And that's what I'm going to show you in the next section. Okay. So we've seen that CRISPR activation in bacteria or bacterially derived systems are sensitive to a number of factors. So we wanted to see whether we could incorporate these design rules with another set of constraints, namely a set of rules for, we want to build now a set of synthetic promoters that we can use to control the expression of anything that we want in response to the delivery of these trans-activating guide RNAs. So that is, we want to be able to express the guide RNAs and build programs that operate through the regulated expression of those guide RNAs. So in this case, now what we're going to do is design this set of promoters. So we want no cross-activation. So we want the guides to target each one of the promoters in an orthogonal sense. We want to have no binding sites for kind of known transcription factors so that these will be interoperable with any other kind of context that we'd like to use them in. And we, of course, want to make sure that they don't have any homology with one another just to make it easier to clone them, essentially. So our initial set had these 4 synthetic orthogonal promoters, and we saw as much as 50-fold activation when we express a guide RNA in the presence of these systems. Again, these experiments are all done in E. coli. So that was a great sign, but we wondered why the other promoters in the set were not able to achieve, why they're not all achieving the same level of activation. If all else being equal, they really should be giving us high levels of expression. So in some of our other work from my group over the years, we've seen that co-transcriptional folding trajectories can have a significant impact on RNA functions, and that a lot of these impacts, the results can best be understood as kinetic effects that are governed by the sizes of the energy barriers that separate the structures of an RNA at the end of transcription from the structure that's needed for function. And so we wondered whether differences in the spacer sequences and the impacts those had in folding into the active kind of scaffold RNA structure could be responsible for the differences that we saw. And so what we did is built 30 different promoter scaffold RNA pairs, all with different targeting sequences and different predicted kind of energy barriers separating this final transcriptional state from the active state that's necessary to associate with DCAS9. And what we found is that the effectiveness of the scaffold RNA directed CRISPR activation does depend on the predicted kinetics of scRNA folding. So these essentially have the same kind of thermodynamic predictions, but this energy barrier kind of separating these two was really the controlling variable here. Okay, so now what we're going to do, we're going to go back and apply our kind of sequence and now structure-based design rules to see if we can expand that set of synthetic promoters. So now when we apply those rules, we were able to create a fifth synthetic orthogonal promoter, a fifth, sixth, and seventh orthogonal promoter that achieved the same levels of expression as J3, which was previously the highest one. And that it gives us predictable and kind of targeted levels of expression. In fact, we've now used this approach to create a set of something like 8 different orthogonal CRISPR-A promoters that we can use to target independently for programmable expression. Okay, before we leave this section, there's one last capability that we want, and that is we want to be able to tune the level of expression that we get without modifying the promoter. So again, remember, we're trying to build these programs that operate through regulated expression of the cognate guide RNA. And so what we reasoned is that if we truncate the 5' end of the spacer sequence, so that is the sequence in the guide RNA that targets that recruits the dCas9 and targets the DNA, that this would reduce the affinity of the scaffold RNA for the engineered promoters, which would reduce the level of transcriptional activation. So now what we're doing is applying those sequence and structure-based design rules to generate a new set of constructs where even when we truncate them, they're still expected to fold. The guide RNAs are still expected to fold. So this is kind of a coupled design problem. But when we do that, we can generate tunable levels of CRISPR activation. So we can now get orthogonal CRISPR-A, build these orthogonal CRISPR-A promoters that are wholly engineered, completely synthetic, to give us independent, programmable, and tunable expression. All right, so that's the set of tools that we wanted. Now, I'm going to show you quickly two demonstrations, or two kind of illustrations. Let's see how I'm doing. That we can use these tools to program variations in gene expression. Again, in E. coli first, and then we're going to move everything to Cellfree in just a second. So, we want to test the new synthetic promoter tools. Can we actually use them to program variations in gene expression, and do they generate predictable outputs? And so the first experiment we did was to transform E. coli with a plasmid expressing a fluorescent reporter protein, so in this case, RFP, BFP, or GFP, under the control of a synthetic CRISPR-A promoter. And then we're going to transform the strains with a combinatorial library of scRNA programs. So there are 3 different scRNA programs that are expected to simultaneously generate high, medium, low, and no expression at all 3 of the engineered promoters. And what we see is that we only see, if you kind of go to the far right-hand side, kind of basal expression in the off for the kind of OT conditions, so off-target guides, and then predictable variations in reporter gene expression that depend on the identity of the scRNA program that's delivered to the cell. So this shows that we really can get the expression of multiple genes to be independently tuned with these multi-guide CRISPR-A programs. Okay, let's see. So before we move to the last section, this is a very simple example or an illustration that is just to show you how we can use these tools to program variations of gene expression in a pathway. And so what I'm going to show you is, so now what we're doing is we placed the expression of enzymes necessary to make lacto-N-tetraose, which is one of the most abundant human milk oligosaccharides, but incidentally also a sugar that's expensive to synthesize at large scale. So now what we're doing is driving the expression of 3 heterologous genes that are expressed under the control of the synthetic promoters that we designed. So the same promoters and cognate RNAs, scRNAs that were used to generate the variations in fluorescent reporter gene expression. And so now what we're doing is putting the CRISPR-A components themselves under the control of a tetracycline responsive ATC promoter. So the whole thing will be inducible. And I'm showing you results from what we might call a kind of a focused library, so a subset of all possible scRNA library designs. And what we see is that when we induce with ATC, we're able to generate significant amounts of LNT, the desired compound, so something like 160 milligrams per liter. But interestingly, we get this significant production only in the low, high program state, which illustrates, and this is consistent with lots of observations in the field of metabolic engineering going back decades, that generally speaking, the highest levels of production from an engineered pathway are rarely obtained from just blasting the expression of all the genes in these pathways. So you really do have to balance the timing and the expression levels of all the genes in the pathway to kind of get the most efficient flux, or the most flux through the pathway. And in the cellular context, this means you need to balance the supplies and demands for cellular resources. As we start thinking about developing these kinds of systems in cell-free platforms, there's a similar kind of requirement that we're going to need to balance the supplies and demands for cell-free resources if we want to get efficient expression of all the enzymes and then have those enzymes efficiently convert inputs into targeted outputs. All right, so let's do that then. So the work that I've talked about to this point has 2 features. The first is it's all been in bacteria. And the second is that the expression components and programs have all mostly been static or triggered in the case of the HMO, the sugar production, but not dynamic. So in this next part, what I'm going to do is show you how our new CRISPR components can be assembled into multi-layer circuits for building dynamic genetic programs in cell-free systems. So, but I want to take a slight diversion here just to kind of motivate this work. So membrane proteins are present in a wide array of cellular processes, from kind of primary and secondary metabolite synthesis to electron transport, single carbon metabolism. But a key barrier to applying or using membrane proteins for applications in bioproduction is that it's really hard to express them and get them to be functional. So not only do they have to be expressed and fold and then insert into the membrane protein activity itself is also highly influenced by the physicochemical properties of the associated membrane. And so this makes it really hard to get high kind of optimal membrane protein performance outside the organism where that membrane protein evolved, whether that's a plant or a microbe. So what we're really interested here is developing kind of membrane-based cell-free systems with phospholipid flavors that we can tailor to emulate specific environments. So say, for example, that we want to take a plant P450 that does a hydroxylation reaction and converts a secondary metabolite into a drug like Taxol or galantamine, an Alzheimer's drug that we're actually working on. And we want to express this in a cell-free system. Then our idea, or kind of working hypothesis, is that we could create phospholipids with component flavors that make it look like a plant cell environment. So we're going to emulate a plant cell environment and use this as a way to express these functional proteins. So we've been working now for a number of years with Vincent Oro at the University of Minnesota and Pamela Peralta-Yaya at Georgia Tech. And with those groups, we're developing these membrane-based cell-free platforms for doing enzyme discovery and pathway prototyping, and then ultimately production of plant natural product drugs. So our collaboration has recently discovered approaches for using membrane-based cell-free systems to produce phospholipids that grow new vesicles in situ. So this is a really important capability for kind of creating a self-assembling system. And we've also seen that plant P450s, which are notoriously difficult to express in microbes, can be functionally expressed in membrane-enabled systems. So our next challenge, or the way that we're thinking about this is to build dynamic genetic control systems that can kind of stagger phospholipid and natural product biosynthesis. And this would allow for in situ liposome self-assembly prior to membrane protein expression, and then following chemical synthesis. And so we think that these capabilities are essential, more or less, for balancing the supplies and demands for cell-free expression resources and ensuring that we can get high levels of functional enzyme expression and pathway production. Now, of course, in naturally occurring biological systems, dynamic gene expression is used to conserve resources, respond to stimuli, generate complex time-dependent behavior. But they're really only limited examples of synthetic dynamic transcriptional regulatory networks that can do anything like complex multi-gene regulation. I think a lot of this kind of paucity of examples can be attributed to the limited number of suitable components for building these networks, and really in the difficulty of combining the components into multi-layered operations. So what we really want to do here is create a scalable framework for rationally designing and tuning dynamic regulatory programs. And then we want to apply this in the immediate sense to the problem of membrane-based bioproduction systems. So, what I showed you in the first part of the talk is that we have these new rules and tools for effective CRISPR activation in bacteria, which suggests it should be possible to do CRISPR activation in bacterial cell-free expression systems. And so we reasoned that if we could combine CRISPR-A with CRISPR-I, then we could access network topologies and functional capabilities that you really wouldn't be able to access using CRISPR inhibition alone. As far as I know, there aren't any examples of prokaryotic CRISPR activation being applied in genetic circuits beyond kind of elementary operations in a single layer, whether in bacteria or anywhere else. So, to kind of go beyond these elementary operations of activation and repression, we formalize this framework for constructing higher order CRISPR-AI circuits that operate through interconnected CRISPR-AI nodes. So in this framework, CRISPR-AI nodes are discrete transcriptional units that have target sequences for CRISPR activation and for CRISPR inhibition, CRISPR-I-directed transcriptional regulation. So, of course, the first thing that we had to do, the first challenge here really, is just to adapt the CRISPR-A system that we developed in E. coli for use in the cell-free expression system. So to characterize each of these nodes, we're going to isolate all the components onto individual plasmids, and then this allows us to do kind of independent titration of the expression levels for all the CRISPR-A and I components. And so what we can see is that if we increase the concentration of the scaffold or sgRNA expressing plasmids, this gives us higher overall levels of activation and impression, as well as a faster onset of the control action. All of which kind of follows this about a 40-minute delay between the initiation of the cell-free reaction and the onset of the CRISPR-AI control, which is consistent with previously reported maturation times of DCAS9 in cell-free systems. Incidentally, what we also found is that there's a really strong correlation between kind of basal promoter strength and promoter activatability of using CRISPR-A in E. coli compared to a cell-free system. And so, this is kind of shows this good correspondence means that we really can use these components that have been developed in E. coli and port them to our E. coli lysate without too much trouble, although we do have to kind of understand how to translate results that we get in bacteria to the cell-free system, but off on the right track anyway. Okay, so if we want to build a multi-layer circuit, then we need to combine, or we built these kind of activation and activation repression cascades by doing level matching between the input and output dynamic ranges. Between nodes. So what we're doing here is matching the output transcription levels of an upstream node that encodes an scRNA to the relevant transcriptional input range of a downstream node. In the case that I'm showing you here, this is so a CRISPR activation in the first layer, CRISPR inhibition in the second layer. So you activate the first one, and this has the effect of repressing transcription from the second node. When we do this, we can see that it's responsive to changes in the input guide RNA span approximately 2 orders of magnitude. Across this range, we can see that the CRISPR-A controlled transcriptional output levels vary by something like 24-fold. And so, the dynamic range of the CRISPR-A generated CRISPR-AI output doesn't fully span the dynamic range of the inputs, but this characterization does show us that the CRISPR-AI nodes can be sequentially combined if we carefully match the upstream output dynamic range with the downstream input dynamic range. That's a little complicated to say, but essentially, if you match the inputs and outputs, you actually can get the signal to be transduced from an input through an intermediary layer to an output. To kind of investigate the ability of these multi-layer circuits to encode dynamic gene expression programs, we compared the relative production rates of, in this case, red fluorescent protein that's regulated just by a single layer CRISPR inhibition to those generated by an activation repressing cascade. And so what we can see is that there are kind of negligible differences in the expression dynamics when if the sgRNA expression levels are mismatched, but if the concentration of the plasmid expressing the sgRNA in the second layer is too low, then we don't get any response. And if we have high concentrations of the sgRNA expressing plasmid in the second layer, then it effectively looks like CRISPR inhibition. It's just it's sort of repressed all the time. But then there's this kind of broad range in the middle where if there's significant overlap between the response curves of the layers in the circuits, then we can get large changes in the timing of gene expression, which kind of underscores the importance of doing the level matching in the first place, but also showing that depending on the particulars of the system, you actually can change the dynamics of the underlying system. So let's make this a little more tunable and rational. So after we establish these rules for kind of governing the construction of layered CRISPR-AI circuitry, we wanted to make some more complicated transcriptional programs, and especially programs that would actually have phenotypes or kind of functional properties that we care about for implementing our bioproduction systems. So now what we're doing is combining CRISPR activation repression cascades with CRISPR activation to build an incoherent type 1 feed-forward loop, so an IFFL, so a classic pulse generating circuit. And so if we take kind of level matching into consideration here, then we can see that the circuit topology determines the timing of gene expression. So we don't see any difference in expression dynamics between CRISPR-A and then CRISPR-A kind of with plus CRISPR-I at low concentrations of the inhibitory kind of CRISPR-I node. But if CRISPR-I is activated by CRISPR-A to form an IFFL, then we can see the gene expression pulse. That's kind of the red and the green going up and then down. That would be characteristic. It's qualitatively different from the expression that's generated by either of the other circuit topologies, and also consistent with our kind of expectation for what an IFFL should look like. We could tune the timing of gene expression pulse generated by the IFFL by varying the concentration of the sgRNA expressing plasmid, so the CRISPR-I plasmid, essentially. We can see that the maximum RFP production rate could happen 110 minutes earlier in the cell-free reaction when we increased the sgRNA expressing plasmid concentration tenfold. More generally, we saw that the time of maximum gene expression pulse could be continuously varied kind of over a fourfold range in sgRNA plasmid levels. So we could shift the maximum the peak of expression earlier by up to 2 hours compared to unregulated expression. Now, what was really important to us is that we could capture the expression dynamics and kind of evaluate the feasibility of rationally designing these circuits by constructing a coarse-grained model. If we use this coarse-grained model and we feed it one set of inputs, then it can accurately predict the shape of the pulse that results from another configuration of the system. And so, what this really does is kind of this observed predictability of the level matching in the cascades, and then these results showing that we can rationally tune them, suggest to us that this really is a way of kind of designing and tuning in silico given component characterization data. So we're now using this framework to engineer dynamic circuits for programming gene expression in these membrane-based cell-free bioproduction systems. Okay, so let's just wrap up, briefly summarize. We've seen that the requirements for bacterial CRISPR activation target sites do pose challenges, but we've also seen that CRISPR activation can be effective at a broad range of target promoter sequences. By systematically defining rules for effective CRISPR-A sites, we've identified new strategies for improving and generalizing synthetic gene regulation. We now have a set of tools and kind of emerging capabilities for engineering multi-gene CRISPR-AI programs in bacteria and cell-free expression systems that are derived from bacteria. So these programs can be encoded as genetically compact sets of scRNAs and sgRNAs. The programs can be used to specify a lot of different functions for activating and repressing genes, implementing dynamic expression programs. Now, of course, we're still learning about these systems all the time, so I don't want to give you the impression that we've solved all the challenges in CRISPR-AI program engineering, but these tools are already giving us some new opportunities for developing these bioproduction systems that can make molecules that we're interested in. All right. So I think I'll stop there and take questions if anyone has them. Thanks again for your attention and the invitation.
Thank you very much, James. So are there any questions? So maybe I will start. It will be very nice if you can comment on how the dynamic CRISPR-Cas transcriptional control system might be affected by the physicochemical properties of the lipid bilayer.
Of the lipid bilayer itself, right? No, that's a great question, and I would say it's something that we have not seen that the expression systems are impacted much by the presence of intact vesicles, but we do expect that that producing phospholipids in their presence may have an impact. But I can't say that we actually know that we have a very clear understanding of how that's or what's going to happen exactly. I think it's a great question and certainly on the top of our list of kind of things to try to understand better. Yeah, I appreciate the question. Thanks.
Thank you very much. There is another question. Thank you for the nice talk. Would there be any advantage in exploring the use of your technology in yeast model system?
In a yeast model system, I guess they mean a cell-free yeast model system, presumably, I guess. Yeah, yes, I think that, so we have, in some other work, we've built multi-layer CRISPR inhibition cascades with Eric Clavens' group in electrical engineering here at the University of Washington that can do Boolean logic and a bunch of other things. So it expresses many as seven guide RNAs in yeast. CRISPR activation is actually an easier task in a eukaryotic system because the rules about how you recruit the activator protein relative to the transcription start site are more forgiving. And so I don't see any reason to think that you couldn't get some interesting and kind of complex behaviors by combining activation and inhibition in that kind of system too. It's not something that we're actively doing, but I think it could be very interesting.
Thank you very much, Professor Carothers. I don't see any further questions for the moment, and we also actually already running out of the time. Thank you very much for your presentation. It was very nice and interesting. So now we will have a break of 5 minutes before the next session. Thank you very much, and have a beautiful rest of the day.
Whenever you want, Professor Lincoln.
Yes. Hello.
Hello?
All good. Whenever you want.
We're good. All right. All right. Well, hi everybody. Welcome to the second session of today, and the last talk session of the conference proper. We've got 3 great talks coming up. Two contributed and 1 featured talk. Without any further ado, I will introduce the first speaker, who is Eleonora Bailoni, who is a PhD student at the University of Groningen in the Netherlands in the Department of Membrane Enzymology. She's also a member of BASIC, which is the Dutch consortium for building a synthetic cell. Go ahead, Eleonora, when you're ready.
Yes, thank you very much, Matthew. Welcome, everyone. Today, I will talk to you about a multi-chamber flow dialysis setup that I have developed for sustainable metabolism in synthetic cells. So we are all here because we are all interested in synthetic cells and in the making of a synthetic cell that can eventually grow and divide over time, hopefully over multiple cycles. And I hope that you agree with me that we do need to develop a sustainable metabolism first in order to then achieve growth and division. Now, to be able to develop this, we need 3 essential components. First of all, we need a compartment where we separate our metabolism from the external environment. However, this is not sufficient. We also need to make the compartment selectively open so that small molecules can selectively be either imported or exported from the external environment to the synthetic cell. And eventually, we also need to take into account that we need to recycle all the internal byproducts that are not exported, because otherwise they would build up and inhibit the metabolism. So we have to always keep in mind all these 3 components when we think of sustainable metabolism. And with this in mind, we look at a practical case study. In our lab, we are really interested in energy homeostasis. We like to make ATP by exploiting the arginine deaminase pathway or arginine breakdown pathway. This is a very beautiful pathway that makes use of 3 cytosolic enzymes, ARC-A, ARC-B, and ARC-C, that convert 1 molecule of arginine together with 1 molecule of ADP into 1 molecule of ATP and 1 molecule of ornithine as a byproduct. And now the beauty of this system is that there is a membrane component to it, a membrane antiporter that imports 1 arginine molecule while at the same time exporting 1 ornithine molecule. So you can really appreciate that we get a double benefit here at the same time, because we get rid of 1 unwanted waste product while at the same time importing the substrate for the next round of synthesis. And now, with this setup, we are able to produce ATP and to accumulate it, build it up inside our vesicles. But of course, it is also interesting to do something with it, right? And I decided to couple ATP synthesis to glycerol phosphorylation in order to produce glycerol 3-phosphate. Now we can do so by including glycerol kinase in the system, and we look at glycerol 3-phosphate in particular because this is an essential intermediate in the phospholipid biosynthesis. That is something that we are also looking at in the lab. So what we do is we prepare these vesicles, we reconstitute all the enzymes, we put the vesicles inside a test tube and start the reactions by addition of the substrates, glycerol and arginine. And we look at the ATP levels over time by means of a ratiometric sensor called Perceval HR. When we do not supply glycerol to the system, we see a relatively fast buildup of ATP relative to ADP, to the point where all ADP is converted into ATP, and then the system comes to a steady state. However, in the case of glycerol addition, we do see that ATP buildup is much slower because ATP is hydrolyzed for glycerol phosphorylation. In this particular example that I give here, all glycerol is converted into glycerol 3-phosphate. Therefore, the ATP levels eventually level off to the final level. And this system is really beautiful. It is really versatile. We can play around with the glycerol levels. We can play around with the arginine levels. But then we stopped for a while and we asked a question that is, we are now working with an open synthetic system that we then place in a closed environment to which we add the substrates at the beginning, but then over which we lose control over time, because of course the arginine is being depleted over time, the glycerol as well, of course, and then the external byproducts also build up. And in our case, ornithine might become inhibiting for the membrane antiporter RD at some point. Of course, this is also a valid point for a synthetic gel more in general terms. When we will have developed a very complex metabolism, we will come to a point where we will want to control the external environment more tightly. And therefore, we ask the question, can we open up the external environment as well, but in a selective way, of course. And by that, I mean, can we retain the vesicles, but expose them to a controlled flow of substrates? And this flow would also be convenient to then wash away the byproducts. So we have given this problem some thoughts over time, and then we decided to look at dynamic dialysis as an option for us. There are really many geometries and possible devices in the literature, but there are three main requirements that we really want to achieve for us to work with flow dialysis. First of all, we want to be able to sample the vesicles, to analyze them offline, for example. And then we want to avoid all sorts of stresses, either on the membrane filter of the flow dialysis, but also on the vesicle themselves. And by taking all this into account, we decided to focus on a dialysis cell that would have a vertical setup. So now we gave it really an extended thought, and we went through many rounds of optimization, but we built our own flow dialysis device that is composed by two compartments. One compartment hosts the cells, and the other compartment allows a feed to flow through. The two compartments are separated at the interface by a dialysis filter. The dialysis chamber is also controlled with a jacket for temperature control. And both compartments can be accessed for sampling or for injection of desired solution. What is really beautiful about this setup is that it is modular. So here you see that 4 chambers were connected in parallel. Of course, you can connect more of them, and you can connect them to different flows as well. So as I said, developing this setup was quite challenging, but we made it and then started playing around with it. Of course, first, there are a few technical questions that need to be addressed. And these are in particular related to the dialysis filter. The first question is, does the dialysis filter really retain the vesicles? This is an essential point. And to test that, we prepared vesicles that contain ADP. We placed them inside the device. On one side, we had vesicles in the presence of the filter. And as a control, we took the device, but we sealed it off with an impermeable sealing so that the vesicular compartment couldn't be in contact with the feed compartment. And now then we looked at the ADP levels over time, and we could nicely observe that the levels found in both conditions, with the filter and without the filter, coincide nicely. So this indicates to us that we do not have a significant vesicle loss over time. You do also see a decrease in the signal, and this is due to a dilution factor that we introduced by sampling. Of course, this is not so much of an issue because we can correct for it. And the second question is, does the dialysis filter allow for equilibration of the small molecules? And what is the equilibration rate that we can measure? For that, we prepared a solution of arginine and ornithine, and we let it flow through the device, and we could observe equilibration after several hours. So now that all the controls were in place, we could finally perform the exciting experiment. We put our vesicles with ATP homeostasis pathway inside the vesicle compartment, and we fed the glycerol and the arginine from the feed chamber. So in the absence of the glycerol kinase, which is the red trace over here, we see that ATP builds up relative to ADP over time in a way that is comparable to our optimized batch conditions. So we were really happy to observe that the flow device can reproduce our earlier data. However, when we did add the glycerol kinase to the system, we didn't observe any accumulation of ATP. And that is because in this particular setup, glycerol is no longer limiting, so ATP doesn't have a chance to accumulate over time. This is also what we see in a control in batch where we provide an excess of glycerol. So this confirms our evidence for the batch experiment. And with that, I come to my outlook. I've shown you that we have developed a multi-chamber flow dialysis setup that allows us to hopefully control the external environment of our selectively open vesicles by allowing for a constant feed of substrates and constant washing of the byproducts. With this, we can perform real-time perturbations, and we can also sample the vesicles for offline quantifications. As a proof of principle, we have reconstituted an energy homeostasis pathway that used to generate ATP coupled to glycerol 3-phosphate synthesis. And we also did not observe an ATP buildup when a glycerol feed was provided. In the future, we would like to proceed in the characterization of this pathway by, for example, quantifying the glycerol 3-phosphate levels. And of course, we would also like to use this for phospholipid biosynthesis. With that, I would like to thank my supervisors, Bert Pohlmann and Arnold Driessen, as well as some other members of my lab that have helped in the developing of the flow device, the colleagues from the membrane enzymology department that you can see here, and all of you for your attention. Thanks very much.
Thank you very much, Eleonora. We have a bit of time for questions, and there's one question in the Q&A already, so if there are any more, please go ahead and type them in. So the question is from an anonymous attendee. It says, how do you control the direction of the antiporter on the membrane?
So the direction of the flow, it means. So what we do?
I'm not sure. My reading was that there was a membrane protein involved. At least that's how. So the question.
Well, there are two things. One is the direction in which the membrane antiporter is reconstituted inside the membrane, and that we do not control. We just assume a random orientation of the antiporter. And the direction of the flux of arginine and ornithine we control by first encapsulating ornithine inside our vesicle, and by providing arginine from the outside. So because only one species is initially present in the vesicle, and arginine will be done on the outside. So there is only one possible way of exchange. And of course, once the arginine is imported inside the vesicle, this is really rapidly converted to the further species in the pathway. So arginine doesn't really have the time to be exported again through the membrane protein, I think.
Cool. Another question from Alan Liu. Does your flow dialysis setup have mixing on the sample side to prevent vesicles from settling?
Exactly. I forgot to mention that, but I can maybe go back to my drawing. Yes. You can apply stirring to both chambers. Yeah, this is a stirring bar. I didn't mention it in the presentation. Yes.
And
so
he also
apply,
sorry, no, go ahead. I thought you finished.
We apply a mild stirring to make sure that the vesicles are not stressed as well. But yeah.
And he also asks, are your synthetic cells SUVs or more cell-sized vesicles?
They are large unilamellar vesicles that have, well, they are extruded through a nominal a filter of 400 nanometers, but of course they come in a size distribution from the extruder. Yeah, they're not giant unilamellar vesicles.
And a question from Tom Robinson says, do you have a nice setup? He says, do you have a control to ensure SUVs don't burst or leak?
So for that, I plan to reconstitute an internal tracer or a membrane tracer. I already have a feeling about it when I performed the ADP experiment, because the dilution factor that I measure is comparable to the theoretical dilution factor that I can calculate theoretically. So if it would be much different, then I would be worried that a large fraction of the vesicles would suffer, because then, at least in the sample with the filter, the ADP would be diluted out. But of course, this needs proper investigation in a way, and I think a tracer is the best thing that we can include.
Okay, excellent. I don't see any more questions, so if there are no more questions, then maybe we can move along to the next talk. And thank you very much to
thank you once more. Yes, thank you.
Okay, so our second talk of the session is a featured talk by Tobias Erb. And he is a microbial biochemist and director at Max Planck Institute for Terrestrial Microbiology in Marburg. And his research centers on discovery, function, and design of novel CO2 converting enzymes from microorganisms to algae and plants, and artificial photosynthesis. He studied chemistry and biology at University of Freiburg in Germany and the Ohio State University, and was also at University of Illinois and ETH Zurich before becoming group lead at Max Planck in Marburg. And he's received many awards, including the Heinz Meyer Leibniz Prize and the Otto Bayer Award. Thank you. So with that, I will pass the microphone over to Erb. Take it away.
All right. Can you hear me? And do you see my screen?
Yes. Right.
Awesome. Great. Thank you. Good morning or good evening, wherever you are on this planet. Hi. Thanks so much for the introduction, Matthew. Thank you so much. It's my great pleasure to present recent research from our lab, in which we aim at rethinking, reinventing, and eventually rebuilding photosynthesis using the methods of synthetic biology. And so what we really try to do is creating new to nature systems that can take up, capture, and convert inorganic carbon driven by light and convert it into living organic matter. And I think this is, for the purpose of this talk, but also for the course of this conference, maybe in a nutshell, what we are trying to do currently in our lab. So of course you can ask the question, why are we interested in this? So what is our central motivation? And our motivation is very simple. It's a very simple molecule that is in the air surrounding us. And it's this small molecule shown here, carbon dioxide or CO2, which you of course know very much as an important greenhouse gas. And I think we all know that, of course, this is an important and central molecule for life on this planet. Currently, it's threatening our life. It's directly related to climate change. And that's typically where we look at CO2 as being really a problem.
At the same time, I think it's also a solution, because it's in principle as simple and eventually the most simple carbon source on this planet. It's easily available. It's basically an oxidized carbon that sits out there in the atmosphere. And I think we as scientists, we as a society, and eventually also any synthetic cell would need to make its living out of it. So the question here is, I want to pose is, can we design new ways to capture this molecule from the atmosphere and convert it into useful compounds for us human beings, but also for any synthetic cell in the future? Of course, you could ask, why do we need to reinvent this process? It does already exist in nature, and you're absolutely right. This process is already existing in nature. It's a process that is driven in biology by photosynthesis. So photosynthesis is nothing else than light-driven capture and conversion of atmospheric CO2 into biomass. And so you could say, well, it's all invented. That's true. But the point is that biology was good at inventing this process, but it was not good at making this process perfect. And this becomes obvious if you look at nature's most important CO2 fixing enzyme, or actually the enzyme in photosynthesis that drives carbon capture and conversion. This enzyme is called ribisco, short ribulose 1,5-bisphosphate carboxylase oxygenase, which is the engine or the entry point, if you want to say so, for carbon dioxide into plant biomass. Well, what's wrong with the enzyme? Well, on the one hand, it is a very slow enzyme. So it takes only 5 to 10 CO2 molecules per second. And this is surprisingly slow compared to other enzymes, which can turn over molecules at 10 to 100,000 per second. So one problem, it's slow. The other problem is that the enzyme makes a lot of mistakes. It makes actually an error with another gas in the atmosphere, oxygen. It actually has an error rate of approximately 20% with oxygen, which means every 5th time, instead of fixing a CO2 molecule, ribisco would take up an oxygen molecule, and this would lead to a process called photorespiration. And this process caused a lot of problems to photosynthesis, and at the end of the day, waste approximately 20 to 30% of the energy in photosynthesis. So apparently nature has come up with a solution that's based on ribisco, but ribisco itself is not perfect. Now, as a synthetic biologist or engineer, you could say, let's simply improve the enzyme, let's try to make it better. Well, many experiments show this is not as simple as we think, because you can, on the one hand, improve the enzyme to make it faster, but on the other hand, you say, so the enzyme would make more mistakes. And vice versa, if you try to make the enzyme more specific, better working with CO2, you automatically slow down catalysis. So in other words, it seems like this enzyme is exactly it's an evolutionary trade-off, and there's not much you can do, at least from a protein engineering point of view. And this really has triggered research in our lab, where we thought, if we cannot improve photosynthesis, most important enzyme, so what about a different approach? So what about if we then simply say, what if we could find, discover, engineer new CO2 fixing enzymes? And what if we can use these new CO2 fixing enzymes to build a complete alternative carbon capture and conversion pathway? Let's say a synthetic CO2 fixation machinery or network, metabolic network. It's of course a bold challenge, and you could ask, is this actually possible? Can we rebuild one of the most evolved and most important pathways in nature? And you might want to take an inspiration from nature to ask this question. So what I show you here is basically the metabolic landscape, the space that nature has explored during the 3.5 billion years evolution on this planet. So you see here is the different CO2 fixing enzymes that we know in biology. And the dots and the nodes that connect those CO2 fixing enzymes are each representing a pathway. And what you see here is that nature actually has invented, on the one hand, CO2 fixing pathways in plants, but also in prokaryotes and microbes, microorganisms. And in total, there has been 7 different CO2 fixation pathways that we know of that nature has evolved over the last billion of years. It's great. You can actually get inspired by these 7 pathways, but you could also see that there's a lot of open space, right? A lot of things have not been tested and not been explored. This is actually now let's say the motivation of our lab, to go in this open space and use synthetic biology to realize those things that nature has not looked at or has not explored yet. And what I want to say here is, this is not a metabolic engineering effort. So this is not a way to basically improve existing pathways. This is really a challenge to invent, test, and realize things that have not been invented so far by evolution. Now, do you want to do this in a very defined way? So first of all, you want to go ahead and design these new to nature pathways on the paper. Those are basically a design and in silico phase. Then we'll go ahead, find the individual parts, the enzymes, the proteins that we would need to realize these new to nature networks. We'd optimize networks in an iterative process. And then ideally, we would like to implement them into a given cellular system. This could now be a natural cell. I'll talk about this at the very end of my talk. But it could also be an artificial cell. And this will be the main focus of the next 20-25 minutes. All right. So how do we design such synthetic CO2 fixation cycles from first principles? So what we do is a process that you might know from chemistry if you're a chemist, which is called metabolic retrosynthesis. So you really sit down and you think from the end of the molecule, from the end of your process, and you redesign a way towards the one molecule you want to synthesize. In other words, you really start to draw synthetic, artificial, hypothetical pathways for the capture and conversion of CO2 on the paper. What you see here is a couple of these examples. There's the catch cycle, the Hopek cycle, the Chyme cycle, the hit-me cycle. All of these are different ideas we had in our mind when we kind of sat down and did this creative exercise. So all these pathways are very different in terms of the enzymes or the potential enzymes you want to use. All are very different in terms of the different metabolites you want to use, but all of them also share some common principles. And the most important principle is that all these pathways are cycles, which means the product of a C2 fixation reaction we imagine is converted back over series reactions to become the substrate of the C2 fixation reaction. And by this, you can run a continuously C2 fixation. So it's one of the common design principles. And the second design principle we always also use is basically a so-called valve reaction. That's a reaction within the cycle where you can split off some of the fixed carbon. You can take this carbon out and you can use it to synthesize an important building block, an amino acid, an organic carbon, a molecule, whatever you want to do with it. So in principle, that's the way to take the fixed carbon out of the cycle and do something else with it. Now, there are many different solutions. I actually show you 5. We have designed many more of those cycles. The question now is, which of these cycles you have in mind is a good cycle? And what are the criteria to evaluate which of the networks you've drawn is a good one? We could also ask if you're a PhD student or a postdoc, which of this gives you a great paper, right? So which one of these you can actually realize in the lab? So you need to come up with certain criteria. And one criterion is, of course, kinetics. We already heard the talks before, how important kinetics is for many of the metabolic networks. So the question is, which of these reactions you draw is a fast and efficient enzyme reaction? And take an example, Rubisco, which I told you is a very slow CO2 fixing enzyme. Ideally, we're going to use a different kind of chemistry. And what we've been using in our lab is really a new principle of CO2 fixation called ECR, which is 10 to 20 times faster than Rubisco. In other words, at the same time, you could fix 10 to 20 times more CO2. So kinetics, speed matters. The second principle that matters, of course, thermodynamics. The question is, how much ATP do you have to spend if you want to fix carbon dioxide? This is shown here. The catch cycle is first design, would use 4 ATP per 3 CO2 molecules fixed, which end up in 1 pyruvate molecule. How much is that? Well, this is approximately 2 times more efficient than natural photosynthesis. So in other words, the synthetic design seems to be faster, at least in theory, and it needs less ATP than the naturally evolved pathways. Last but not least, there's more criteria, something like thermodynamic feasibility. So we want to operate along a free energy gradient to make this really a very efficient process. Well, the point is, this is just theory so far. So now it's really about building these pathways in the lab. And this means you need to go back into the wet lab, you need to find the individual parts, the enzymes to build the cycle at the end of the day. To make a long story short, we really don't care where we get the enzymes from. We just screen databases, we test different enzymes, and then we identify those that are capable of doing one or the other step. And to build the first version of the cycle, what we really had to do is bring together 15 different enzymes from 6 different organisms and 1 engineered enzyme. And at the end of the day, this really allowed us to build the catch cycle, realize the catch cycle in an Eppendorf cup within one defined condition. Well, having isolated all these enzymes, found all these enzymes, the question, of course, then was, if you now put all these enzymes together, we add ATP and NADPH, energy that we would need to run the system. And if we add 1 metabolite of the cycle, this green compound down here called propyl-CoA, would the cycle start to turn, and would it actually convert CO2 into the output molecule, in this case malate. And yes, indeed, we could show that the system starts to turn and it starts to accumulate malate over time. This is great because it tells you you can really think of a new to nature pathway, draw it on the paper, analyze it, go back to the lab and identify and find the parts, engineer one of the enzymes even, and you can build a metabolic network, and it does actually fix carbon dioxide. Well, this is great, but we also realized that at the end of the day, the system was quite slow. It was far from being very efficient. And actually it fixed only 0.2 CO2 molecules per acceptor hour, which is really, really way less than we expected. So the question really is, what keeps the system, this metabolic network out of 15 different enzymes, up from running well? And the answer is very simple. We already heard in the talk before, and actually in James's talk, and that it is all about getting the enzymes efficiently work together. And what we have seen is that many of the enzymes that come from very different biological backgrounds have a problem in working together. There are some metabolites which just get lost from the cycle because of side reactions. Other metabolites might inhibit some of the enzymes in the pathway. And there's also crosstalk. So there's a lot of things that should not happen. The network is not working perfect. And now it's all about optimizing the one metabolic network we've built. So you basically have to play evolution. You have to debug the system. To make a long story short, we use different principles. One is, of course, enzyme engineering. We design active sites so that some of the reactions become better. We use detouring, which means certain problematic reactions we completely leave out. We use other chemistries to do the same reactions. And then one interesting and important principle is actually recycling or scavenging. As soon as we see that a certain metabolite gets lost from the cycle, we can actually then try to recycle it, add more enzymes to the cycle, make the system more robust, and basically create something that you could call scavenging. All right. So building more complexity to make the system more robust seems to be a principle that's actually some of really importance if you build metabolic networks. Make a long story short, you implement all these different principles and you can ramp up activity of the system by almost a factor of 20, which means you do not have to really rethink the initial system, but you have to optimize individual parts. And this is already enough to increase the system's efficiency of the system by an order of magnitude. So at the end of the day, the system is now with 17 different enzymes from 9 different organisms and 3 engineered enzymes. So way more complex, version 5.4, which tells you also how many different rounds of, let's say, test, improve, build, test, analysis we had to go through. Right, this is of course not the end. You can ask, can you even systematically employ the optimum and improve the system even further? As you can imagine, it's a very complex task. It's many different components, many different variables, many different parameters you would have to test. And what you've now started to do is kind of mimicking evolution by using machine learning, active learning. So the idea really is you build many different versions of the catch cycle. You vary the protein concentrations, you vary the metabolite concentration, you vary cofactor concentration, maybe also the pH. We test them in a high throughput in a small scale fashion. In this case, we use an automatic pipetting robot, and we work in a microliter scale. We analyze the output, which of the system works best, and then we feed it back into an active learning algorithm, and we try to improve in the second, third, and fourth round, of course, the system towards an optimum. What you can see here is indeed that these assays and these approaches seem to improve the system. This is actually ongoing work. I know that Chris, who might be listening to talk, has run the 5th round today. So this is really data generated while I'm talking. And what you can see is starting from round 1, where we had in varied different conditions, we continuously improved the average of the output. We even have a couple of experimental conditions where the system seems to be almost a factor, don't get me wrong, almost a factor 10, even better again than in the initial round. It's great. So machine learning can really help optimizing such complex systems by active learning. It can also help in another way, identifying factors and interactions that seem to be important. So certain elements that need either to be, that work complementary, or that you need to kind of have a strong link in improving or making the system worse. And so machine learning, active learning can also help you to identify beneficial interaction dependencies. And we hope really to to learn about the system and in a fundamental way using these new approaches. Okay, so improving the system with active learning is one thing we're currently working on. Another thing is, of course, now building even more complexity. So what I've shown you is a system of 17 different reactions that convert CO2 in a molecule called glyoxylate, a simple C2 compound. But of course, that is not what life is built on. Life is built on many more different complex compounds. So the question is, can we now take this glyoxylate molecule and can we now convert it into, diversify it into much more different products? So can we convert glyoxylate through another metabolic module, another metabolic module 2 into an acetyl-CoA, for instance, which is an important precursor to build many different compounds, for instance, terpenes, which you know as flavors, fragrances, biofuels, or pharmaceuticals. To make a long story short, we have played around and we have really now redesigned a system in which we could take glyoxylate as the output molecule of the catch cycle, then feed it into a natural module called the beta-hydroxyaspartate cycle that we had rediscovered recently in marine proteobacteria, to convert 2 molecules of glyoxylate into 1 molecule of oxaloacetate. And then we use, let's say, part of a citric acid cycle to go to acetyl-CoA. And this acetyl-CoA then we diversify through mevalonate pathway in different terpene synthase and different compounds, in this case, limonene, sabinine, alpha-pinene, and even sesquiterpenes like bisalobinene or beta-farnesene. What you can really see is that in the end of the day, if you combine the catch cycle, the 17 reactions, with these approximately another 15 or 16 reactions in one Eppendorf cup, you can really build those molecules directly from CO2. We've done another exercise, which is going into the direction of polyketides, which you know as antibiotics, fungicides, or anti-cancer drugs. The question was, can we also build these molecules starting from CO2? Again, you see the network gets even more complex. This is now on the order of about 50 to 60 different reactions you couple in one Eppendorf cup. And indeed, what you see is you can really also build these complex systems, build complex molecules from CO2 in a modular fashion, combining all these different modules together. And we really produce polyketides, for instance, this compound here, which is the building block of erythromycin, from CO2 directly. Okay. Well, I've been talking so far about making complex molecules, right? So what I want to switch to, the second part of my talk is now telling you about not building complex molecules, but building molecular complexity. So it's a little bit different task, a little bit different challenge. And this is basically inspired by the fact that many biological systems are highly structured in time and space. So take, for example, natural chloroplast, which is the CO2 fixation machinery inside of the plant cell. So if you look at it, it is really well structured. It has thylakoids, which are photosynthetic membranes that are stacked. It has parts where the enzymes are concentrated. It's of course operating in day and night cycles, and it's dynamically regulated and organized in time and space. So ideally, in an artificial cell, an artificial chloroplast, if you want to build such a system, you'd also hopefully be able to create a similar level of complexity, and you will be hopefully also able to achieve the same level of control in space and time. And this was the challenge of one of the latest projects in our lab. So how can we build such a system that is able to fix CO2 with light? Well, there's different challenges you have to overcome. First of all, you need to be able to now run the synthetic catch cycle, not with ATP, not with NADPH, but you would like to run it in directly using light as energy source. How can you do that? Well, one simple example and approach would be you take existing chloroplasts, you try to extract the photosynthetic membrane, so-called thylakoids. This is what we did here. So we went onto the market in Marburg. We basically bought spinach. We grinded up the spinach and we carefully isolated the photosynthetic membranes, these thylakoids. What you could show is that this isolated thylakoid fraction can indeed convert light into ATP and can convert light into NADPH. So with isolated thylakoids, you can create the energy and the reducing covalence required to do metabolic conversions. And then what we did is taking those photosynthetic membranes, this natural photosynthetic machinery, and brought it together with our artificial CO2 fixation cycle. What we try to do is optimizing, so powering the catch cycle, the synthetic cycle, with the natural photosynthetic machinery. Is it possible? Well, what we have learned is, in fact, it is not possible. So apparently, the natural photosynthetic machinery and the catch cycle do have issues with each other. In other words, again, if you try to bring two modules together that have not evolved together, you run into problems. Then it's all about finding out what the problems are. So why does the catch cycle in version 5.4 not operate very well with photosynthetic membranes? To make a long story short, it comes down to basically two enzymes that cause problems. And one enzyme is methylsuccinylco oxidase. It's an enzyme that creates reactive oxygen species. The other enzyme that makes problems is called PCO. It's a propyl-CoA oxidase. And again, this enzyme creates reactive oxygen species. And what you should know, and what you probably know, is that, of course, photosynthetic membranes are very susceptible towards such radical oxygen species. So in other words, while running the cycle together with the photosynthetic membranes, you create and destroy the photosynthetic membranes. And what you need to think of is now a way to kind of overcome this problem. What we did is basically replacing MCO, this oxidase, by dehydrogenase. And what we did in the other case is replacing PCO, propyl-CoA oxidase, by propyl-CoA carboxylase. So we create a version 6.0 and 7.0 of the catch cycle. And once we implemented and used these enzymes together with the synthetic membrane, you could see that, in fact, those two systems, those two modules, the energy module and the CO2 fixation module, work together, and we could really produce glyoxylate, or in this case then glycolate, in a light-driven fashion. Right. Well, so far what we did is just doing a bulk experiment, but ideally we'd like to have a system that mimics the chloroplast. You would have a system which you can confine CO2 fixation to a certain compartment. And for that experiment, we've teamed up with Jean-Christophe Barré, a microfluidic engineer in Bordeaux. It was really great collaboration. What we have done together is building, let's say, micro droplets using microfluidics to create water and oil droplets that we can then equip with photosynthetic membranes and with our enzymes, basically building a kind of a mimic of the artificial chloroplast. What you see here is one of these examples where we basically have encapsulated enzymes together with photosynthetic membranes. And what you see is that if you shine light on these droplets, they would start to produce ATP and NADPH. What you see here is NADPH fluorescence. And as soon as you turn off the light, you have enzymes that would consume NADPH. And then NADPH is basically taken away. The fluorescence goes away. You can recharge again by shining light on the droplets. And what you see on the right side panel here is you can really charge up the droplets with light, turn it off, NADPH is consumed. You turn on the light again, you charge up the droplets. And so in a basic light on, light off fashion, you can recharge the droplets for, let's say, approximately 2 hours. And after that, they start kind of losing in activity at some time. But of course, it's not only about not just charging up and charging, basically using up the energy. It's at the end of the day, of course, can you bring in more complex cycles? And what you see here is, in fact, when you bring in the catch cycle inside of these droplets, you can really fix CO2 into glycolate. You can do it in a light-driven fashion. You can do it even as, if not a little bit better than if you did it in bulk solution. All right. So I think this is was very exciting for us. We called it the artificial chloroplast. It's in principle, if you want to translate it into a bit more scientific terms, it's an enzymatically active system confined to a micro droplet that is at the interface of the natural and the synthetic biological world. Natural, because you still have these photosynthetic membranes built in the system, and synthetic biological because we really have completely redesigned CO2 fixation machinery in using synthetic biology. But of course, you can ask how much synthetic can you get? So can you basically extend the system? Can you go even towards more towards material sciences? So So if you think about the natural chloroplast on the left side being completely biological, and think about photovoltaics, the light-driven creation of electricity of the other side, could you create maybe a system that could use electricity, but still do biology? So can we have some electricity-driven biological CO2 fixation? And this is a current ongoing project in our lab, where we really want to use electrons to power metabolic pathways. This is done in a 3-lab collaboration, where the idea really is you have an electrode that delivers electrons into a redox polymer. And this redox, these electrons can be picked up by enzymes, in this case, ferredoxin NADPH reductases. These electrons can be handed over and put onto NADPH, and then you can actually run classical biology, for instance, reductive carboxylation driven by NADPH consumption. What you can see is if you build these systems, indeed, they are quite active, at least over 24 hours. And we can see if the systems really can, in a light and electricity-driven fashion, convert, for instance, cotonou-CoA, shown here in purple, into ethylmethylcarbonate fixed carbon dioxide, picking up the electrons from the electrode. Well, that's great. And I think that's the starting step for building more complex systems. And what we would like to do in the near future is not only bring one single C2 fixation reaction onto these electrodes, but we would actually run the complete catch cycle using electricity at one point in time. And this would basically now bring biology and electricity together in a very interesting, very exciting fashion, at least for us. But at the very end of my presentation, what I would also like to do is now again going back into biology. So I've talked about the natural chloroplast, I've talked about an artificial chloroplast, I've talked about bioelectrical systems. But of course, one question is, once you've built these really new to nature synthetic pathways, is there any chance that you could get back them into a living cell? And as James Carothers already pointed out, integration is not a trivial task. It's not very trivial, because what you need to do is basically take in a system of 17 or 18 different reactions and put it back into a living cell that features more than 3,000 different reactions. And you can anticipate that this is, of course, a technological challenge. You need to express up to 17 or 18 different enzymes in a controlled fashion inside of a living cell, for instance, E. coli. And it's also not trivial, because you, of course, have to think about all the potential interferences that can happen if you bring in a new to nature metabolic network, an existing metabolic network. Again, substrate that might be inhibiting, side reacts that might be ongoing, stalling of the cycle, poisoning through different metabolites. So the question really is that possible, and are modern cells flexible to adopt towards complete new to nature networks? So we've taken on this task, and we have basically tried to teach E. coli to use the catch cycle to make a living out of it. So how do we do this? Well, what we do is we basically isolate a certain metabolite in the metabolic network of E. coli. In this case, succinyl-CoA, which is a central carbon metabolite in the citric acid cycle. So we knock out all different pathways, all different routes leading to succinyl-CoA. And the cell can then not grow. It cannot grow unless, of course, you provide it with succinyl-CoA, or you provide it with new means to synthesize succinyl-CoA. So this is so-called auxotrophic strain. And you see, indeed, if you grow the wild type, the cell does not grow. If you have the selection strain, this knockout strain, the cell is basically dead unless you provide the precursor in this case. Now, what you can now do is bringing in all the reactions of the catch cycle. And this is what we have been doing. We have brought in 80 or 90% of the reaction of the catch cycle. And we then have basically fed this compound here crotonate. The idea, if crotonate is fed into the catch cycle, you actually create crotonate-CoA, you have the carbon fixation reaction, and all the way down into succinyl-CoA, which is then provided from crotonate and CO2. And indeed, if you express all these, I think in this case, 12 or 13 enzymes inside of E. coli, you feed crotonate, CO2, you see that the strain starts to grow, and it really can use now this artificial network to make its essential precursor and divide. And I think that's the first step towards now adopting and implementing this new to nature network inside of the living cell. And of course, what it opens also is a possibility of improving now the interaction of all these different enzymes with each other. In other words, This might be an alternative way to our artificial intelligence efforts to improve the network, to design, build, test, and learn. Here you would have a strong incentive, the strongest incentive ever. It's natural evolution. The cell should grow faster. It can only grow faster if it starts to evolve the network. So let's see which of the two approaches is better. The natural one inside of E. coli, or the biological intelligent one inside of E. coli, or the artificial intelligent one where we use machine learning and lab automation. All right, so that's the end of my presentation. I just want to make one small disclaimer. What I've been trying to tell you is that we are building new to nature pathways that are more efficient than those that are used by nature and have evolved by nature. But the point is not to save the world with the small droplets or the C2 fixing E. coli, either or the bioelectric systems. Right now, it's more about really getting insight how to build such systems, how to make such system robust, and how to build molecular and modular complexity. So it's really this fundamental insights to design and build these systems that drives our motivation, our research. And of course, we hope that at some point in the future, we will be able to also apply the systems in a large scale efforts. But right now, it's really about building those artificial chloroplasts and the CO2 fixing natural and synthetic cells. I hope we could show you that it's really possible to design new to nature features, new to nature metabolic networks on the paper. You can really realize them then in the wet lab. You can optimize them, hopefully using artificial intelligence in the future. Then also, of course, I hope could show that we are about to really put them inside of natural, but also artificial cells. And this is maybe really the challenge for the next 5 to 10 years in our lab. Final remarks. I think we can really, at this point in time, design and build new biological solutions based on chemical physical logic. We really need to understand a bit better, though, how to biologically design or build in biological design principles into our design. Proofreading, channeling, compartmentalization seems to play an important role, and this is something we need to address in the future, also with our synthetic biological efforts. And of course, it's also all about finding these enzymes or engineering these enzymes to build these new to nature networks. So de novo enzyme design, databases, large-scale screening will help us and build a database of potential new building blocks we can use in the future. And the last but not least, the most exciting one, of course, the tools and methodologies to build natural synthetic cells and integrate such complex metabolic networks in the context of these cell or cell-like systems. I'd like to thank my team. It's a great team in our lab. So I just want to thank the biochemists, Iria, Gabriele, the people who developed the catch cycle, Taryn, Thomas, the complex molecules team, Chris, Patrick, Vidya, the people that try to electrify the catch cycle, David and Shanshan. And last but not least, the genetic engineers that try to go back in vivo, Alberto and Hai. I thank you for your attention. And Matthew, I've seen you turned on your screen. I'm happy to take questions. Thank you.
Yes, thank you. Very interesting talk. If you do have any questions, go ahead and type them in. There is one there already from RP Oates, who says, this is very interesting work. Thank you for your presentation. I'm wondering how good these metabolic cycles are at accumulating end products. In other words, do you find shifts in equilibrium constants upon reaching higher concentrations of an output molecule such as glycolate?
This is a very good question. Indeed, what you want to have is out of equilibrium systems. And what we typically see is when you run these cycles is we start to accumulate a metabolite. And in certain cases, the metabolite is either unstable and starts to decay, right? Or it starts to inhibit the system. And so what will be really important for us is to have a strong sink. So what we typically try to do is have a strong thermodynamic endergonic hexagonic reaction at the very end, where we basically drive the system towards a state and we pull out the product. So you can do this, for instance, by in the future building a polymer. Or you can do it if you think about now a synthetic cell that you kind of export the product. And this is also what a natural system is used. The chloroplast does CO2 fixation inside and it pumps out the fixed carbon. So one of the challenges in the future will be to build GOVs, which would have pores and you could pump out your product constantly. And this would keep the system out of equilibrium. So this is really one thing that is really important to realize in the future for us. Mm-hmm.
Yeah, I had a question if anyone else has questions, feel free to type them in. I guess one thing I was wondering is sort of related to that Is there any benefit, for example, to have sort of control of the product to maybe, you know, divert resources to different products in response to, you know, conditions or demand?
Of course, this is also a really great question. What we like to do is to build a flexible network that can react to different conditions. So what we like to be able to do is, for instance, creating different products. Now you can either think about doing this in a controlled manner through an outside signal, or you can hopefully let the system itself sense its let's say its energy status and make decisions. So I think this is something we're really up to. And what we have been trying to do is creating multiple outputs. So let's say one thing is asterisk A, but then we also think about taking away parts of the cycle and diversify and diverge. And so we can do it either completely on the protein level to allosteric interactions and to let's say tuning the kinetic parameters and have feedback loops, or in the future taking transcription translation to, you know, one product accumulates, which induces a second set of let's say of enzymes which are produced, and then you start to diversify this product. So this is all things that are possible in the future, hopefully, and we are really interested in building such more complex, reactive, responsive networks in the future. Matthew. Yeah.
Cool. And there's a question that's coming from an anonymous attendee who says, thank you very much for the nice presentation. How do you break the natural metabolism and couple your designed pathway into E. coli?
Wow, I hope I don't break E. coli at the very end, right? So it still needs to live. So the point is, what we have been doing is, of course, taking individual purified enzymes to build a system in vitro, right? So now trying to put it into E. coli is challenging because you really want to basically redesign central carbon metabolism. And typically metabolic engineering, you kind of branch off and you have a side reactivity and you branch off the cycle. Here it's really complicated. And this requires, on the one hand, let's say smart design, thinking about the metabolic network of E. coli, at the same time also figuring out how much orthogonal you are and how much side reactions are ongoing. So this is really an ongoing effort. And you basically put in small pieces, try out if they work, and then you build more complex things in E. coli. Mm-hmm.
And a question from Mihir Dingankar says, thank you very much for the interesting talk. Do you have any strategy to mimic protein turnover in synthetic systems, as otherwise the proteins would lose activity after a certain number of cycles?
Yes, that's the holy grail, of course. We already heard about how to build and to replace and substitute things that break. I think that's the thing why, for instance, our photosynthetic system breaks after 2 hours, right? So in a living cell, you always have constantly replenishing of those parts of the photosynthetic membrane that just die, right? So for us, it's two ways to do that. One thing would be, for instance, to make enzymes more robust, and that's basically an optimization function that's been used very often in biotechnology. If you think about laundry powder, right, we have enzymes in there that are stable towards heat, towards pH differences. So that's something we could do in the future. The other would be to have transcription translation, right, also in a controlled fashion again. If the system senses it breaks, it starts to repair only those parts that break. And this would again require smart sensing to some extent. But I think in the future, this might be one thing we would like to address also in one or the other way. Yeah.
Great.
And of course, what you can also do, sorry, I forgot to mention this, is if you think about systems, and it's of course also compartmentalization. So let's say there's certain chemistries which are not compatible with each other. So what about, let's say, concentrating those enzymes that are compatible with us in one part of the artificial chloroplast, use for instance liquid phase transition, or another way to encapsulate, and then the other part of the cycle into another compartment. And this is also what nature does, right? So in a way, building in space, kind of controlling the network in space is also very important for us. Yeah. But again, the tools need to be developed. It's not that easy right now. Mm-hmm.
Thank you very much. If there are no more questions from the audience, then I think we will thank Dr. Erb for